Okay, now it's recording.
Yep.
Shall we start? Yeah. Welcome, everyone. So yesterday we had a great session and hope to see going to have another great session today. So I might repeat the same thing what we had yesterday, just introducing our team behind this work, amazing team, and Ane from Singapore, Sling, Biswa from India for Envada, and Fahana from Agri Research. I always say it's an Institute of Plant and Food Research Limited. Professor Urek, you always helping, always from the Singapore for Munich, and also Professor Tianen go from Westlake University, and myself from SAIC. So we are going to put that program to support everyone in research in proteomics, metabolomics, and lipidomics. Today we're going to give 2 awards as yesterday for the best lightning talk and the best runner-up, worth €250 for the best talk and the €100 for the runner-up. Sponsored by Journal of Steroid Biochemistry and Molecular Biology and the Westlake Omics. Sorry, Mike. So around the clock, we're going to provide the best for everyone in the globe. So hopefully we can cover most of the countries. Those who are missing, we're going to stream the recording in around 8 o'clock Singapore time. If you miss, you can have the video later. I will upload into YouTube. Today's schedule is fully packed with very nice talks, 3 keynote speakers, 5 lightning talks, and 2 tutorials. So without delay, I would like to introduce our first keynote speaker today, Dr. Jenny Cooney. So thank you so much, Jenny, joining. I know you had a busy week. Jenny is a chemist with the Plant and Food Research Biological Chemistry and Bioactive Groups. You are from New Zealand, and you are going to talk about phytohormones, your area, and your title is Phytohormone Metabolomics Platform using mass spectrometry to understand plant signaling. Really looking forward to your talk, Jenny. Floor is yours. I'll stop.
Share my screen. Thank you, Susie. So, as Susie said, I'm a coordinator for physiological chemistry at Plant and Food Research. So Plant and Food Research is a Crown Research Institute in New Zealand, which was set up to enhance the value and productivity of New Zealand's horticultural, arable, seafood, and food and beverage industries, with a mission to create the world's most sustainable food systems. So our metabolomics platform is very diverse as a result. The scientists working in projects.
Jenny?
Yes.
Can you please click your display settings? Click your display settings in the top.
Yeah, the swap.
Yes.
Yeah, swap.
Click.
The first one.
Duplicate. The second option.
You want duplicate?
The second option.
Yeah. No.
Yeah.
Okay.
All good now. All good now.
Yes.
Yeah.
Okay. So we have scientists working in projects from fundamental plant research through to applied industry-related projects and nutritional-based clinical trials. So our metabolomics platform is predominantly based on mass spectrometry with a small NMR component. And in common with many other research institutes, we have specialist mass spec equipment and skills for untargeted metabolomics, targeted metabolomics, and imaging. So I'll be talking today primarily about our targeted metabolomics. first of all, I'll give you a few brief examples about untargeted and imaging capability. So first of all, an apology to my GC mass spec specialist colleagues. Today I'm going to be focusing solely on the LC mass spec platform. It's highlighted 4 of them here in our slide, one of them you'll recognise. They predominantly work in the flavour, volatiles and taste space. So please do contact the HANA or the conference organisers if you're interested in knowing more about the type of research. So our untargeted EMS capability utilises high resolution CMS instruments, but it also has ion mobility capability. And although the workflow is set up primarily as a global screen to identify metabolites and priori, which may be up or down regulated between different data sets, they do of course have the ability to reinterrogate the data to look for specific metabolites and give relative quantitative information. And this type of workflow requires minimal sample workup, but relies heavily on statistical analysis and data filtering to give useful outcomes, such as markers of disease, disease management monitoring, novel trait discovery, or breeding targets for germplasm screening. So untargeted metabolite analysis gives a lot of data. It's very hard to get an overview of the screen, so the project, so visualization becomes a really important tool. So on the PCA plot here shown, this is the work on PFR's apple metabolite germplasm collection, and each of these dots represents a different apple from the germplasm collection. And so on the left here we have greybean, which is a sweet dessert apple, and on the right we can see golden hornet, which is described as a crab apple. It definitely wouldn't be described as being sweet. And so you can see that most of the apples in the collection are clustering to the left. That's likely a reflection of the breeders selecting for sweetness as an important trait in the breeding program due to consumer preference. So what's underlying that? Well, looking at 4D metabolite landscape plots can help visualise the clinical differences underlying that PCA discrimination. And so we have along the x-axis, retention time, the y-axis we have accurate mass. The bubble size represents the intensity of each metabolite, and the colour gives us an indication. And similar compounds, class compounds, will have a similar aspects of the south and side. And so we can see for the greybean sweet apple, we have quite a lot of terpenoids, where not surprisingly for the more bitter tasting apple, we have a lot more polyphenols and the sugar organic acid, which gives us our sweetness perception is also altered. So in this next example, Nigel was looking at fungal markers of disease in grapevine woody trunk disease, and he's using a volcano plot to represent the metabolites and show the data. So within this experiment, he was looking at the unique fungal metabolism markers for Palata and Salmonella fungal diseases identified in and it's comparing the entire metabolome and then comparing those to control healthy tissue. And so on the plot, the circles on the right of the plot are upregulated in the disease tissue relative to the control. Those on the left are downregulated. And as I move across the plot, the fold change increases. So the pink and yellow circles indicate the metabolites that are unique fungal markers specific to a particular pathogen. So unfortunately, in this case, these markers were localized to the wood tissue, which was not particularly useful as an on-orchard disease marker progression tool, because it means you have to actually dig up the plant to be able to identify the disease. So they were actually able to identify some markers in the leaf tissue, and hopefully in the future and they'll be able to develop a sensitive protocol device to use on orchard for early detection of these type of diseases. Now, an imaging example. The targeted and untargeted metabolomics tells us what metabolites represent, but mass spec imaging tells us the where. So using this approach, Andrew was able to segment tissues into chemically similar zones without prior knowledge of the tissue structure or targeting any particular ions to generate a segmentation map of the developing bilberry in its blue unripe stage right through to the ripe expired stage, and generate the segmentation map developing bilberry. And in that way show that the ripening process proceeds from outside to the mesocarp. And this was performed in both negative and positive mode, which detects right classes. So each coloured region of the map represents a cluster of single spectra, allowing chemically similar regions to be defined. So if we look at the negative mode data first, you see the arrow here, that little blue area that's progressing from the exocarp. That was identified as a particular cinnamic acid. And then as ripening proceeded, we see the emergence of flavonols, and then by the ripening stage, there in evidence right throughout the carp. In positive ion mode, we see the emergence of anthocyanins, and as development proceeds, the anthocyanins right throughout the mesocarp. And they can combine these images to construct time-lapse videos, which are very, very cool. So now we move on to structural targeted metabolomics. And so not surprisingly, as a plant-based organisation with an extensive breeding program, many of the research programs we are involved in are focused on fundamental research. So we've developed this phytohormone metabolomics capability to quantify multiple classes of phytohormones, as indicated here. And we look at about over 50 phytohormones, including precursors, conjugates, and catabolites. And that means we can look at biosynthetic pathways rather than just active metabolites. So this is based on the triple quad platform with Q-track functionality with a couple of ABSI instruments, 500 and 500. And we use labelled standards for accurate quantitation. It is a unique research capability in New Zealand, and it's contributing to improved understanding of hormonal control, plant development, and fruit, apple, carrot, berry fruit, amongst others, and of course model plants for hypothesis testing. So why are phytohormones important? Well, phytohormones underpin all plant responses. So similar to humans, where hormones are the body's chemical messages sending signals into the bloodstream and tissues to regulate processes, including growth and development and metabolism, plants rely on plant hormones and phytohormones to orchestrate plant responses. And different phytohormones control different aspects of plant growth and development and stress responses. And then you have considerable crosstalk amongst the different classes to fine-tune these responses. So, for example, if we look at flowering here, we can see contributions from gibberellins, auxins, ephyrin, and cytokinins. Some of these interactions will be synergistic and some will be antagonistic. And then just to complicate matters, light, environmental challenges and physical challenges all can affect the status of phytohormone balance. And then, let's say, for example, the plant has to out of defense against the pathogen. There's also an energy trade-off in that, and that will affect plant growth. So this is just a snapshot of some of the types of phytohormones we look at in our research. And we have two separate workflows depending on the type of experiment looking to focus. So for experiments investigating plant stress responses to various challenges, we have a separate stress phytohormone screens we call it. So that uses about 10 to 100 mass of fresh tissue for a dry weight equivalent. We do it all on the 96 well plate format, and we'd extract them twice So then go through a carbon graphite plate cleanup, and then we'll be looking at salicylates, jasinates, ABA, first type compounds. And this is just a few of the metabolites in the screen, on the screen. For experiments where knowledge of plant growth and development is also important, we have a fuller screen, which is a lot more labour intensive, so a little bit more tissue, and we also extract 4 times. And then the metabolites go through a cation exchange plate, and this separates out the cytokines into a separate phase, and they go on to the next step. And the remaining metabolites we look at all have a carboxylic acid function. So ADA, the gibberellins, the jasinates, the salicylates. And so after they go through the cleanup rate, we derivatize them to increase the sensitivity for analysis for the gibberellins, because they're present in plant tissue in very, very low quantities. And so we use MS probe and stabilized protein, which is just fancy way of saying we derivatize them. And that has several advantages. So first and foremost, it's the sensitivity. So we take this as a gibberellin and simple structure. Carboxylic acid functionality is derivatized with pharmacoline. And so we get an analyte as a result, which ionizes a lot better in the next step. So it flies better, we get a much bigger response, and so our sensitivity of analysis is greatly increased. It's also useful for specificity, because then you get a very characteristic fragmentation in the next step. And so if you want to do some different experiments where you're looking for related unknowns, that can be very handy. And then the real big advantage is the generation of labelled standards. So compounds such as gibberellins, there's not a labelled internal standard for every analyte commercially available. And so we can take our analytical standards, and in a one-pot reaction, we can derivatize those with a labelled probe, so the labelled equivalent of bromocholine, and in doing so, generate an internal standard for all of the analytes that we're analysing for. And by using the ratio of the XP to the internal standard EXP, we can use that for quantitation. So now I'll just give you a couple of examples where we've applied. In this first experiment, we're investigating the link between phytohormones, carbohydrates, supply, and fruit size. And this is important because consumer acceptance of fruit is determined by size, flavour, and lightness. And in this study, we investigated how altering carbon supply, the carbohydrate supply to food and fruit, altered the balance between growth and accumulation of metabolites. So we used leaf plucking and girdling to manipulate the carbon supply in the leaves to developing fruit. So fruit on the vine were thinned and leaves were plucked to give a leaf to fruit ratio of either 2 leaves per fruit, which is would be considered low carbohydrate supply fruit, or 6 for high carbohydrate, and more control in between. Then the canes were girdled, which means putting a fine girdle, which means putting a cut around the cane, to prevent the transport of carbohydrates from the leaves, which is the source of carbohydrates, to the roots of the seed, and at 2 time points over the developmental season, which in here are described as early or late. So we then monitored fruit growth, metabolite accumulation, cytokinin concentrations, and maturation, and the sensory attributes of the rock at this time point were assessed and compared with control fruit. So you can see here at ripeness that the final weight of the early high and late high carbohydrate fruit was greater than the control. And the high carbohydrate fruit had increased starch, sugar, and cytokinin concentrations, and began to mature earlier than control fruit. They were also perceived sweeter This is just a trace of transient and life cycle, which is the major cytokine we detected. And if we looked at all the cytokine data, we could see from the PCA analysis that leaf supply, the carbohydrate supply, leaf density was more important on fruit size, it was controlling level. There are the high carbohydrate data sets, early and late firdling, and low carbohydrate ones are on this side of the plot. So that was more important in time. So we also were able to show that the zeatin type cytokines were most significantly positively correlated with fruit size. So we did another experiment using red kiwi fruit, where we looked at a wider class of phytohormones and also transcriptomics and put them together. And that confirmed that the cytokine concentrations were most tightly correlated with fruit weight and carbohydrate supply. And that changes in many of the other phytohormones were actually mostly developmental and not correlated. The cytokine and biosynthetic genes and expansion genes were also downregulated under carbon starvation. And unlike in other fishy fruit where IAA and gibberellins drive food expansion, in kiwi fruit this seems to be limited. Suggesting a new role for cytokines in kiwi fruit that contribute to stimulate cell expansion by expansion proteins. And in this last example, investigating the link between phytohormones colour and ripening in blueberry. And so, unlike in the bilberry example they showed earlier, where as the fruit ripened, the pigment cyanins progressed in concentration from the mesocarp, from the excarp to the mesocarp, and the skin and the flesh became purple. As blueberries ripen, the skin changes from green to purple as the concentration of cyanins increases, but the flesh remains green. So in many non-climateric fruit, the transition from growth to maturation is primarily under control of ABA, which can induce mid-transcription factor expression and therefore anthocyanin biosynthesis. So we hypothesised that the lack of anthocyanin biosynthesis in blueberry flesh would correlate with reduced ABA biosynthesis in the flesh. However, when we looked at that, and so the flesh is shown in grey here and the skin in black, across the maturation series of blueberry, there wasn't a lot of difference in the concentrations, and neither did they correlate with ABA concentration, with anthocyanin concentration, which with the skin increased exponentially across the development series, within flesh remained low and constant. However, when we looked at the biosynthetic pathway, the story became clearer. So metabolites and genes associated with ABA metabolism and reversible conjugation were up to 10-fold higher in fruit skin and did correlate strongly with the anthocyanin. This was giving rise to the tissue-specific pigmentation. And it's a good example of why both sides of the biosynthetic pathway is important, not just the quantitation of the main ingredients. So where do we think targeted metabolomics fits into the biometabolomics and multi-omics space? We think using targeted metabolomics as a response to treatment is a powerful tool for linking and interrogating targeted omics approaches. So think of targeted metabolites such as regulators and bioactives and biomarkers as a central dial, pivot around which the global changes revolve to biome systems biology. But of course, this is not a trivial task. And at PFAR, like many research institutes, will see the value in integrating multiple omic streams of data as part of the systems biology approach to better understand and manipulate complex biological systems. The amount of data that these different analysis streams generate and the complexities involved in integrating them to generate biologically relevant information does require specialist tools and data frameworks. So at PFAR, we're now lucky that we can dedicate a team integration. An integration group is tasked with doing exactly that. And so they've developed this framework using R, and it handles pre-processing of data sets, statistical integration, and then develops visualization of best practices and recommendations for future projects. So for us, it's still very early days using this framework, but the preliminary data we're now generating from some new Growing Futures programs is looking really exciting. So I anticipate some very good publications coming out soon using this framework. So watch the space, and that's probably a good note to finish on. So thank you very much for your attention, and also thank you to our working team for joining this Tanya. Thank you very much.
Thank you so much, Jenny. It's a really, really great talk and very nice information there about fruit and unbelievable. I was like, wow. It's
Perhaps
really
I'll
nice.
have time for one question. Or maybe none.
Is there any question in the chat? I might check. Anne, do you want to ask the question or?
I can. Hi. I know nothing about plants apart from how yummy the apples taste, but I was wondering, you showed the metabolome of, I think, Braeburn versus something, and I was just wondering, because in the humans we see a circadian rhythm, and we see when we do longitudinal analysis, we see all sorts of changes. Do you have that?
Yes.
How much do you have that in plants?
Yeah, you do. That's another confounding factor, and particularly important when people are sampling, that they sample at the same time of day. Because yes, there's definitely a circadian rhythm, and it's quite a bit of work going on at Quantum Food around that and using that to exploit faster growing samples.
Okay, cool, thanks.
I would like to ask a question because I'm really fascinated about blueberries, and so it's shown like it's when it's ripe in all that, it's the purple color is only in the skin, but the concentration is not changing. If we eat the whole fruit, it's okay. Still we can capture all, or
yes, I mean, there's goodness in the skin and the flesh, but the anthocyanins are concentrated in the flesh, in the blueberries, not the skin.
yeah.
There's other phenolic compounds, flesh, whereas in the blueberry, the anthocyanins.
Is there any other?
Throughout the fruit.
Yeah, Jen, is there any other metabolites changing with that, or only you look for that?
Yes, no, there's a lot, but that's and that's more the untargeted approach.
Okay, okay.
And so no, there's definitely publications.
Okay, looking forward to that.
That
Yeah, fascinating. Any questions from the audience? Can I ask one more about the apple? So is there any fungus or something growing we can detect from the taste? Have you matched any of the flavonoids or the other metabolites changing with the taste, or other data to see whether it's correlate, or?
now, which one was this with? Was this with the grapevine ones?
Yeah, that's the one. Yeah.
Yes, so I think we'd have to check with Nigel on that one, because I think some of that is maybe IP driven, if they're going to be looking to commercialize anything.
Yeah, yeah. How it's correlate with the flavor. Yeah, so many questions, but I don't want to. I'm looking forward to your publication coming soon. Thank you so much, Jenny. We can move into our lightning talks now. I'll share my screen. So it's my great pleasure to introduce our first flight in talk. Sorry, my laptop is a bit slow today. So Aline Martins. Aline, if you are, I think you join. She's going to present about metabolomics insight in CCC or IDC, metabolic pathway through metabolomics-based mass spectrometry, because her time zone, she's going to present as the first speaker. She's from Script Research USA, Flo is yours. Aline, I'll stop my screen sharing. Please share your talk. Hello.
Hi. Can you listen me?
Yeah.
Okay.
We can see it, but you can put in the presentation mode. Yeah.
Yes. Okay. So thank you very much for the invitation. I thanks to Biswa and to you, Tuzi. And I'm sorry because it's a little late here in San Diego. So I asked even to Tuzi to change my speech because it's already like 9:30 p.m. here. And as you can see in California, there's a lot of sun and we wake up really early here. So, so sorry for the change, but I will do my best to present to you this work. So today I will talk to you about the work that actually I didn't perform in the scripts because now I am at the John Yates lab working with single cell proteomics. So next time when if I am invited, I will talk about single cell proteomics. But today I will talk about a translational work that we have been doing between Brazil and Spain on Chagas disease, which is a very burden disease in Brazil and is growing actually in other places. So the title is Metabolomic Insights in Chronic cardiomyopathy and idiopathic dilated cardiomyopathy, molecular pathways through the metabolomic. As I said, this is a huge collaboration work between hospitals in Brazil and between my home university, which is UNB, the University of Brazil, able to perform this metabolomics work at Coral Barba's lab. So I will talk a little bit about Chagas disease for those who doesn't know. It's caused by
You're breaking.
as you can see.
We couldn't hear you. If you stop
It's okay
your
now?
camera, yeah, probably it might help us. Yeah.
Oh, okay. But it's not supposed to be like this. Just a second.
I can hear you very well. Jurek Adamski speaking.
You do? Yes,
Okay.
it does.
That's good now. Yeah.
You're all good. You're all good.
Yeah, it's good.
Oh, okay. Okay. But if somebody else is not hearing me, because I can't, but the internet is pretty. And this is because factors is spreading out to other places, northern places. So about 30 to 40% of the patients with Chagas disease dysfunction due to the parasite tropism to myocardial cells. So what happened is in the end stage of the disease, the only curative chance is if you do a transplant, a heart transplant, because the diseases hit so hard on the patients that this is the only curative chance that he has. So the the Chagas disease, the clinical manifestation has a worse prognosis in all the cardiac diseases and causes dilated cardiomyopathy, and causes also arrhythmias, thromboembolism, and complications, which can bring to a sudden death. So it's a severe disease and refractory heart failure. So because of this, that is so important in those places that there are a large number of patients with Chagas disease. And this disease leads to a cardiac remodeling, results to a synergetic effect between the immunological, inflammatory, autonomic, and microvascular factors. So CC, the Chagas disease has a worse clinical prognosis if you compare to another heart diseases, because it leads to malignant arrhythmias, kidney dysfunction, hemodynamic instability, which can bring to patients to utilize a pacemaker, resynchronizer, or in the last moment, in the worst outcome of the disease, it can bring subtle death. So the most interesting about this disease is that the younger patients had a shorter survival. So it's a big burden on the economic activity society. So because of this, that we try to approach in a translational matter, doing like a translational research on this subject, which is a bench-to-bedside research, bringing clinical research, clinical data, and epidemiological trials, the knowledge that is generated by this clinical research, and joined with and linked with the basic research, which is provided by genomics, proteomics, metabolomics, I'm sorry for the typo, and to now we are beginning to see the first works on single cell omics, especially proteomics, which bring us to mixture these different ideas to provide a better understanding about the disease. So the first on this experiment that this work that I'm bringing to you today, we evaluate several patients in different scenarios to try to understand a little bit more about the disease. So we use a control patients, which was the donor, the heart donor patient. So this is the best control that you can have, because this patient, the heart of this patient will be transplanted for the Chagas patients. So he can have another underlying disease, but he does not have any kind of heart disease. So we consider this the best non-pathological control. So we compare to all the patients that had dilated cardiomyopathy or heart failure, and we see that on the OPLSDA, the metabolites that discriminates these two phenotypes are very well apart, and the model is really good to discriminate, of course, these two patients. This was expected, but with this data, we could understand what kind of metabolites are involved in the remodeling of the heart. So it was even though it was expected, these differences, but now we understand what kind of metabolites are linked to this heart failure. So the other evaluation that we did was control Chagastic patients, but another kind of patient that was idiopathic cardiomyopathy patients. These patients die of heart failure, had to transplant the heart, but it was not because of Chagastic disease, or not even a ischemic disease, was an unknown disease that causes this condition. So again, we can see that this, when we compare these three models, we have very interesting discriminant metabolites for each one of the phenotypes. So again, we increase the knowledge about the metabolites that are involved in each one of those phenotypes. So the last scenario, and there was the scenario that most interested us, was the Chagastic versus the idiopathic, because both, you need to remember that both of them die by heart failure in very young age, but one of those has a diagnostic, which is Chagas disease. The other one does not. So by this OPLDA, we can also see that there are differences, but the differences are not as big as when you compare the controls. And we expect this because the outcome of both of the disease are the same, which is the failure of the heart. So, but again, by doing this, by reaching this approach, we understand what kind of metabolites are involved in each one of the phenotypes. And the most impressive thing was when we discovered that the metabolites that were discriminant for Chagastic disease were involved in the carbohydrate metabolism and carbohydrate with carbohydrate conjugates derived from mitochondrial redox impairment. This is really interesting on the point of view of metabolites, because recently in 2021, one of our colleagues published a data that a proteomic-based metabolomics experiment that shows that the mitochondrial behavior was impaired on Chagastic disease. So this metabolites impairment also agree with the same findings that our colleague that made an independent experiment found. So to finish the presentation, we did a signaling pathway interactome between all the patients with heart failure and the control. And for the metabolites that were intense and discriminant on this scenario, we find out that the most impaired signaling pathway was again the TCA cycle pathway, which leads to the impairment of the mitochondria impairment. And when we compare the CCC, the Chagastic patients with against the idiopathic patients, we find out that myo-inositol was the most important pathway that was impaired in Chagastic diseases. And this is really important because most of the metabolites that are linked to myo-inositol, it plays an important role on the calcium signaling, which is an important mechanism in remodeling of heart disease progression, leading to arrhythmogenic effects during ventricular hypertrophy. So these metabolites are linked to a known clinical phenotype on those Chagastic patients. So take-home message, the study of the impairment on metabolites and pathways may help to elucidate the molecular pathogenesis of Chagastic disease and understand the origin of discrepancy in their metabolic signature. The future integration of these discriminating metabolites with already widely used clinical data, such as HASI or Seattle score, which are very used in clinics, can increase prognostic capability and organ allocation for these patients. This work can strongly contribute to identification of the gaps in the molecular pathogenesis and monitoring of CCC and provide molecular basis for further clinical decisions. So I would like to acknowledge the people in Spain and Sembio from Coral Barbas, the hospital that provide, all the knowledge is about the patients, the people from Brasilia University, which is my home university. And thank you very much. I'm sorry for the time.
Thank you so much, Aline. What a great presentation. Is there any question from the chat? Anyone want to ask a question?
I think that we have some questions here. So I can hear fine. Okay. I think
please,
that's
if you
just
see
about
some, because of the time, we better move on. But Aline, if you are around, please, if you see any question coming up, please feel free to answer. But
okay.
so
Thank you very much.
Thank you so much. And I'll share my
See you.
screen quickly. See you. So our next lightning speaker, Palkin Arora, she's from Institute of Nuclear Medicine and Allied Science, India. Palkin is going to present systemic metabolomics, discovering tools for traumatic brain injury in rats. Looking forward to your talk, Palkin. I'll stop sharing. Floor is yours.
Hi. Hello, everyone. I'll just share my screen. Okay. Is my presentation visible?
Yeah.
Okay. Hello, everyone. Myself, Palkin Arora, and I'm a research fellow at Institute of Nuclear Medicine and Elite Sciences, DRDO. And I'm pursuing PhD from Department of Biochemistry, Punjab University, Chandigarh. Please bear with my bad throat. And so the title for my today's talk is Systemic Metabolomics, a deciphering tool for traumatic brain injury in rats. Okay, so to simulate a human traumatic brain injury, we use modified Marmara weight drop TBI model in rats, where a weight is dropped onto the rat's head. So the height of the height from which the weight is dropped decides the severity of injury. So if it is like around 25 centimeter, it is mild injury, and if it is from 50 centimeter, it is moderate injury. So our basic question of research was to understand that how injury-induced allostasis affects the hypothalamic pituitary adrenal axis and its outcome on systemic metabolism. To perform our study, we grouped the rats into mild and moderate injury groups, and then collected their serum and brains for histological analysis at post-injury day 1, day 5, day 10, and day 30. And then we clubbed day 10 and day 30 into chronic time points. So our hypothesis was to look at how cellular changes in the hypothalamus actually leads to the release of corticosterone from the adrenal gland, and corticosterone being a pleiotropic hormone, its physiological functions must be affected. So we looked onto the inflammation and metabolism. So moving further to our first objective, so we looked onto that how the microglial cells, which are the immune resident macrophages, how their activation was affected after injury. So this slide shows the photomicrographs of hypothalamic IBA1 stain sections at all the time points after injury. So what we did was we obtained the single cells from these photomicrographs, and then we did morphological analysis on these cells, and we found that the normal microglial morphology changed into an activated state morphology. So this was done using Fiji, and the parameters we performed included somal area, which was found to be increased at all time points after both the injuries, while only decreasing at acute time point of mild injured rats. So this could be due to the proliferative state of microglial population after injury. Then the second parameter was the density of the cell. So density is actually the cell shape. It was also found to be altered to be increased at acute time points of both mild and moderate injured rats. Then moving on to the cell size, which was denoted by span ratio, it was observed that the span ratio was decreased at all time points after both the injuries. And then the fourth parameter was the fractal dimension, which denotes the complexity of the microglial processes. So the complexity increased at acute and subacute time points after both the injuries. So collaboratively, the morphological parameters suggested that a microglia have transposed to a hyperactivated cellular form, and this could actually promote an inflammatory and oxidative environment in the hypothalamus, and thus leading to an impaired hypothalamic function. So therefore, the second objective was to look that does it result into alteration in the corticosteroid release from the renal gland, and it was observed that there was an increase in the cort levels at all time points after injury. So this suggests that this persistent elevation in cort can actually impair the HP axis function. So therefore, we first looked onto the systemic inflammation, where we observed that the serum pro-inflammatory cytokine IL-1 beta levels were increased at subacute and chronic time points after moderate injury, while the TNF alpha levels were increased at chronic time point of moderate injury. Interestingly, the anti-inflammatory cytokine IL-10 was found to be depleted after mild injury at acute and chronic stages. So this suggested that an altered systemic immune response is present after TBI, and it is distinct in both mild and moderate injury. Further, we looked onto the metabolism, where we did serum metabolomics using NMR. So in the NMR, this is the NMR spectra, we integrated 18 metabolites, and then after the multivariate analysis, we found a complete segregation between the control mild and moderate injured groups at all time points. And the VIP score plot, the metabolites in the VIP score plots was used to understand that how systemic metabolic changes were there after injury. So we first quantified the body weight of rats, because it's very important to understand that how their feeding habits and appetite is affected after injuries. So interestingly, mild showed a significant decrease in the weight drop after acute time point. So and also this was correlated with their decreased feeding habits, and along with this we also computed ratios of the important metabolites, where lactate to pyruvate ratio was increased at acute and chronic time points after moderate injury, and at chronic time point of mild injury. Then metabolic perturbation was observed, where alpha to beta glucose ratio was increased after acute and subacute time points of mild injured rats. And similarly, there was an elevation in TMO to choline ratio at acute and subacute time points, again in the mild injured rats. So We also calculated Pearson correlation, and we found that both this increase in the ratios of alpha to beta glucose and TMA to choline ratios, they were positively correlated. So we conclude that after the mild injury, there could have been a release of alpha glucose in the process of gluconeogenesis from the systemic organs like intestines, livers, or kidneys. And there could actually be presence of a possible relation between pertubed glucose, choline, and TMAO metabolism after trauma. So this summarizes my study, where we have found that an activated deramified microglial morphology actually leads to an increase in serum corticosterone levels with a depletion of anti-inflammatory cytokines after mild injury, while an increase in the pro-inflammatory cytokines after moderate injury, with a distinct metabolic profile after mild and moderate injury. And metabolites like lactate, choline, branching amino acids, they have helped us distinguish mild and moderate injury from the control rats, while the alpha to beta glucose ratios and TMA to choline ratios after acute and subacute time points helped us classify mild injury from the moderate injury. Therefore, systemic metabolomics has helped us know the difference between the metabolic functions that happen after both mild and moderate injury. So this work has been published in the journal Metabolomics. And I would like to thank the organizers for giving me this opportunity for this talk. And I would also like to thank my supervisors, Dr. Richa Trivedi. She's a scientist in DRDO, and my supervisor, Professor Ajit Sandhi from Department of Biochemistry, Punjab University. And my team, Dr. Kavita Singh, who is currently working at Harvard Medical School, and one of the research fellows at INMAS DRDO, Megha Kumari. Thank you so much.
Thank you very much, Parkin. What a great story. And about your presentation, is there any question in the chat? While I'm waiting, Parkin, how did you normalize the data when you see that weight is changing with the brain tumor, brain injury?
Brain injury, yes.
And how do you see the weight also changing? Did you use the weight as a normalization factor, or how do you?
No, we just checked the weight of the rats. That was it like pre-injury, we took the weight, and then after injury.
Oh, okay. Yeah, is there any question in the chat? Please feel free to answer because we are running out of the time now. So catching up. Thank you so much for a nice talk, and looking forward to your publication. So now I'll share the screen. Yeah, I'll hand over to Tianen to introduce Professor Marcus. Tianen,
Thank you.
voice yours.
Thank you, Ceci. It's a great pleasure to have our keynote speaker, Professor Marcus Rauser. He's a world leader in high throughput proteomics in the world. And he has been a group leader at the Francis Crick Institute, London since 2013. He recently became Einstein Professor of Biochemistry and is the head of biochemistry at the Charity University in Medicine. And previously, he was the recipient of Wellcome Trust Research Career Development Fellowship and the Wellcome BET Prize at the Department of Biochemistry and the Cambridge Systems Biology Center, University of Cambridge. He has very impressive technologies, including hardware and software for implementing high throughput proteomic analysis. His title of his talk is High Throughput Proteome Technologies for Systems Biology and Systems Medicine. Let's welcome Marcus. Thank you.
Thank you very much, Tianan, for the nice introduction. I'm not allowed yet to share the screen. Duzi, could you activate this for me, please? Also, my camera is not yet active. Yeah.
I don't see him in the panelist. So probably you need to first move him from the attendees to the panelist.
I did promote.
Yeah, the microphone appeared, so the microphone seems to work, but what does not work yet is screen sharing and activating my camera.
It's lucky we can hear.
Because you are a panelist, do you see the green the share screen button, Marcus?
No, there is no share screen button, and there is no camera button. And if you email me the link again, that I can directly jump into the panelist room, then I could quit this one and go in again.
This morning I emailed latest one, but I will do that again.
Did you? Okay. Okay, I can click on this link again.
Sorry for the technical issues. I believe he will be back soon.
Yeah.
We
Yeah, this looks better. So the typical bug fix of any software, just reboot it. Fantastic. Good morning. Thanks, Tianan, for the nice introduction. We share a little history when Tianan was a postdoc at ETH in Zurich, and I was visiting there very often. And ever since, we had great interactions and great contact. Great. So let me share the screen now, finally. Now I just need to click the right button. Very good. You should be able to see my screen now.
Yes. Go ahead.
Fantastic. Let me make this a bit smaller. Great. So I will talk a little bit today about two motivations why we do high throughput proteomics, and both connect a little bit to one of the biggest streams that we share in the biosciences. And this is to solve this genotype to phenotype problem. No? So ever since Watson and Crick, now some 60 years ago, discovered the coping mechanism, or had a model that was able to postulate the coping mechanism of DNA, this dream was born, that eventually we could have a genomic information for an individual and know which phenotype it is. And this over the years initially became plausible, because research found out there is this almost linear looking flow of information from the genome to a transcriptome to a proteome. And we know that in disease, many proteins can be mutated, can go wrong. And we were hoping that this is a solvable problem. No? Now, it is a solvable problem, but 60 years later, we still need to admit that we are not that far in many aspects. Now, we can sequence now genomes, we can determine transcriptomes, we can measure proteomes, but there is still not the knowledge there that we could give, the genome sequence of an organism or of a cell or of another system to anyone, or a transcriptome or proteome, and be sure what the phenotype of the cell or this organism would be. And that, of course, would be highly desirable to achieve, because if we understand this process, we could develop better drugs, we could know what goes wrong, we could improve biotechnology to make a more sustainable economy, right? So there is still a lot of idealism and potential in getting better. But why is it so difficult? So one of the things, of course, is the size of the system. Now, the genome is quite huge, transcriptome is huge, the proteome. But the next step is also quite important. So the phenotype, actually, of an organism, of a cell, is actually less determined by this linear flow in the early steps of our genetic machinery, but a lot by the interactions between the different molecules. And there is one key player here, which on this slide, which I took from the Khan Academy, is just here painted as some side players. No? It's small molecules, it's metabolites. So it's the metabolome that strongly determines the phenotype. And the metabolome is not in a one-to-one relationship with the proteome and these underlying layers. It's in a many-to-many relationship, because most of the enzymes, they produce multiple molecules. They are made up by multiple molecules, and they interact by multiple molecules. molecules. Yeah? And in order to address this problem, we need quite big data sets, high quality data sets that we didn't have so far available in our discipline. So my lab works a lot on this interaction here between the proteome and the metabolome, but I'm not going to talk about this today because this is a bit a technology-driven meeting, but I would like to give you some teasers at the end, but to tell you a little bit about the technologies that we have been doing. Great. So one of the problems that we have in the genotype-phenotype problem is that we still don't understand a large fraction of the proteome. Yeah, and so even in organisms like yeast, you know, which is probably the most studied eukaryote because its importance in biotech and because its utility as a model organism for systems biology, we have absolutely no idea about 1/3 of the proteins, what they do, and we have some scant idea about, you know, another third of the proteins, and we have only good information about a small fraction, 10, 20, 25% of proteins. So there is a historical bias behind this. You know, some proteins kept being studied because we started to study them early on, and we still don't fully understand what they do. And then that's the reason why then people keep using, you know, working on the same proteins. You easier have publishing papers, you get them easier to resources, you have more constructs, more mutant lines available. Yeah? But very often that's the only argument. Yeah? Very often we focus our research on a lot of proteins just because they have been studied a lot. Yeah? And that's of course not good enough because that means we miss gene function, we miss drug targets, we miss the ability to solve the genotype-phenotype problem. Yeah? There are two news pieces from this year which reflect that people know that this is a problem, and our field is trying hard to push for a better understanding of the entire proteome. Now, proteins are of course best studied if we can measure them, and that's why proteomic technologies are important. And the proteomic field is very mature these days. There were huge developments over the last 20 years, but most of the developments in proteomics were driven by the typical applications of molecular biology. Yeah, you wanted to know the function of an individual gene in one context, and this very often meant that you wanted to have long lists, go very deep into the proteome, find perhaps many post-translational modifications, but usually the sample sizes to do this were relatively small. This is because that's the way how molecular biology operates, okay? So you do a cell line, you do a knockout, or you have a mouse line and you make a knockout, or you give one drug or one condition. So deep proteomes necessary, but not big sample series. And this is a great approach for a lot of applications, but some applications you cannot study in this way. Yeah. And so when it comes to systematically finding unknown genes, unknown patterns, predictive models, then we need to take another approach. Then the depth of the proteome becomes not the most important feature. The reason is that biological systems are organized in networks. And when you deal with networks, you don't need to quantify each node or each connection each time. Okay, you can deal with a sparse data set as long as the data set is consistent. But what you need is large sample numbers. Okay, and so my lab has been thinking, coming from a metabolism background where proteomic depth was not the biggest of our problems already for quite a while. How can we accelerate? How can we generate methods that allow us to quantify the proteome in large scale? And this is a long story short here. We had to address every single step of our pipeline, which starts from sample prep over creating triptych digests, over the acquisition schemes, over the software to facilitate this. And just one quick word about the sample prep itself. There are multiple ways of doing it, but what you need to be very careful when you work in large scale is to avoid batch effects. Proteomic and metabolomic techniques are quite susceptible to batch effects. They easily emerge without you noticing. And if you have those batch effects and you do a big study, then of course you might come to conclusions which are entirely technical. Yeah, so be aware of the sample prep. We have published our protocols. Other people have done too. What we have then been thinking a lot is how can we accelerate proteomic data acquisition? Yeah? And one of the big game changers for us was when Rudy Aebersold and colleagues started to make data independent acquisition techniques applicable. Yeah, so SWAF-MS was one of the buzzwords for us, but this meant, you know, that we can accelerate proteomic experiments without having a stochasticity problem or without having a big problem in missing values, which is a problem here. And so we have after this came out, started to work with different manufacturers of mass specs to change a little bit how we do data acquisition, and we started to use faster and faster chromatography. Initially, a few years ago, we switched from nanoflow chromatography, which was very successful in proteomics because of the good sensitivity it gives, to higher flow rates because we could accelerate the gradients and we had less problems with batch effects over long measurement times. And meanwhile, we do a lot with analytical flow rate chromatography. So one of our standard methods now uses an 800 microliter per minute flow rate and 3-minute gradient time. And this, of course, made huge demands on the mass spec side, because even if you have a nice chromatography which nicely separates your peaks, if your peaks are very short, very sharp, then you need a technology which is able to capture those. Yeah? And this gave rise to a series of new methods. I'm just introducing briefly two of them, Scanning Soft and OxoScan-MS, and they can now suddenly deal. And this allows you with a conventional mass spec to increase the throughput in proteomics to something like 15, 16, or even 1800 samples per week, yeah? At a high quality, at a very nice reproducibility. Another good side effect of analytical flow rate proteomics is the costs. The costs go down because the costs in mass spectrometry are mostly driven by the instrument time and less by the consumables you have, which means the faster you measure, the fewer your individual measurement costs. Yeah? So this becomes highly attractive to apply in applied settings, in industrial settings, and in medical settings. And I would like to give you one example of a medical application at the end of the talk, right? Good. And another thing we struggled with is data analysis, because there was lots of great software available at the time. We started to do fast DAA measurements, but each of those software packages struggled with big data sets. Some of them were just too slow, and some of them would just crash. Yeah, and so we started to develop our own software suite for data independent acquisition. In the end, it was named Diane because we use deep neural networks of a very crucial step in the workflow there. But the main motivation initially of Diane was just to have a software which can take thousands of samples and process them. And meanwhile, of course, with all the experience these workflows gave us, Diane has matured and does some other things quite well. And some of you in the audience which do high throughput proteomics might have actually used it meanwhile. Okay, so two teasers about how you can accelerate proteomic experiments. So one is the acquisition scheme, and one method we used or developed is called Scanning Soft. So Scanning Soft bases on a method which was promoted years ago by a company called Waters, called Sonar. And what it effectively does is it replaces a window-based acquisition, that you have in a typical DAA experiment with a scan. It's a continuous scan of the first quadrupole. And the first thing this does is allows you to change the window size independently of the cycle time, which means you have a new optimization of ion flow through and methods, and you don't lose time by focusing on a particular signal or a particular mass range, yeah, or on a particular mass window, which means you win speed. You are faster. Yeah. And the second thing what it does, it adds a new dimension. That's quite important. So when a precursor comes off your chromatography, it is hit by this scan. Yeah. The precursor enters the scan and exits it, and this gives you a new chromatogram type dimension. And this chromatogram type dimension is identical of your intact precursor and your MS2 space, and that means you can map now the two on top of each other and can assign accurate precursor mass to your MS2 fragments. And that allows you to deconvolute much more convoluted spectra that you can get when your work measure very quickly. Yeah, and so this allows us crazily fast. This is just an example from our first publication. This is a 30-second active gradient on an 800 microliter per minute chromatography and a mammalian cell line. I think it was K562, and they quantify here something like 1500 protein groups very nicely, very reproducibly, even after fast chromatography. And if you give us a little bit more time, some 3 to 5 minute measurements, we reached at the time some 4000 protein groups nicely and perfectly quantified. And meanwhile, we are quite higher. And so the difference between a much slower, nano flow rate, much more distorted chromatography, and these high flow rates becomes shorter and shorter, and meanwhile is only 10 or 20% or so. So we can now scale proteomic measurements. Okay. Another thing, and this is very briefly because I don't show many results in this short presentation here, is that we can now use the scanning dimension, this MS1, MS2 assignment, to also call for post-translational modifications. Because post-translational modifications have been not an easy task in data independent acquisition, precisely because you only had information about the MS2 fragments and not necessarily an assignment to an intact precursor mass. Yeah, and once we have this available, actually this works pretty well. And this is an example of a new method that we have just pre-printed. It's called Oxoscan MS, and Oxoscan searches for diagnostic ions, for oxonium ions. That's where the name comes from. That fall off when glycan or glycosylated peptides fragment. Yeah, and so we capture those oxonium ions pretty well in our scans because they are low molecular weight and we can detect them very sensibly, and we can record glycosylation maps. So this is a human plasma sample here. It's a microflow chromatography with a slightly longer gradient, a total runtime of some 45 minutes. Yeah, and these dots here are oxonium ions that we can quantify and identify, and we can map those oxonium ions in one of those plasma samples to about 1,000 glycosylated peptides. Yeah, and so this is now getting also working with faster chromatography, and it's very sensitive, and without any enrichment of the glycans, we can now quantify them pretty nicely. And just a little proof how nice this looks like. So these are really the glycosylated peptides, because if you treat the same sample here, if a mix of glycosidases which cleave off these post-translational modifications, you see that all of those signals disappear. Yeah, so it's a pretty specific and nice method. Okay. Cool. So now we have high throughput proteomics. What can we do with it? And I'll show you one example from the basic sciences that addresses this problem that we still don't know the function of many proteins. And then in the end of my presentation, I'll give you an example from the clinic. Yeah. Okay, a lot we do in basic sciences is done in yeast, and this has three different reasons. The first one is that we can grow yeast very effectively in the lab. We can manipulate it, and we have very nice genetic libraries. And one of the key libraries we have is a knockout collection where every single non-essential gene is deleted, and we can they are bar-coded, the strains. We can handle them in pools, but we can also handle them individually, grow them, and do systematic metabolomics and proteomics for them. Yeah, and so we typically do things in a minimal growth medium because we study metabolism most of the time in my lab, and this means around 4,500 yeast strains can grow. Great. The second reason we work with yeast is that biotechnology loves yeast. Yeah, and the biotechnologists want to know how to manipulate a strain to produce more of whatever compound, and that's pretty important because our economy needs to change, and we need to use microorganisms more and more for producing stuff in an environmentally friendly way. And the third reason that's important because yeast represent pathogenic fungi quite nicely. Now we have a global rise of antimicrobial resistant and tolerant fungal cells. Candida albicans is a key pathogen to humans, but there are some new ones like Candida auris, which really create headaches. And yeast is very close to them, and the better we understand single fungal biology, the easier we can deal with those. So very good reasons to work with yeast. Okay. We did set up protocols where we can grow them systematically and automatically prep proteomic samples from those yeasts. It's a semi-automated way. Semi-automated means there is still a manual supervision of the workflow, but actually everything is done on liquid handling robots. And early on, we decided on a compromise. We used a microflow chromatography with a total runtime of about 30 minutes. And this was still done on a 6600 triple-TOF from SciX running swath and scanning swath. Yeah. Great. Now we combined the two things and we created a matrix here of proteomes. So we quantified the proteomes of all of the 4,500 knockouts that go in our condition. And after very strict quality filtering, we consistently detect some 1,900 proteins in these 4,500 knockouts. This doesn't sound dramatic, but it's actually as good as it can be, because this is about half of the expressed yeast proteome. And as you know, in proteomics, we have a little abundance bias. So once you quantify half of the proteome, you can more or less assemble all sorts of networks that you can possibly imagine, because you capture proteins from every single molecular pathway that is known today. Okay, so this is our matrix, and it links effectively every single gene deleted in the genome, which is response on the proteome. And there are just some data here which shows that this works with this technology quite nicely. This is one of the key ones for us. With these high flow rates and this data independent acquisition, and with Diane, we achieve a very high data completeness. And this was one of the weaknesses of the classic proteomic techniques, which really focused on depth, but where data completeness was never an issue because you just measured 3 or 10 or 15 samples. When you measure thousands of samples, this is key. And so we get a complete matrix for most of those 1900 proteins that we see consistently. Yeah, and you see here in this PCA that we have batch effects under control, so all of the batches don't swap in the principal component analysis. Okay, so very briefly, how can we now use such a data set to assign functions to genes? So of course we can do the classic thing. Now we can look at individual proteomes for the knockouts and say if these proteins get upregulated, that associates this protein to this particular pathway. But we can do other things which are new and nice and give you more statistical power. One of them we call reverse proteomic profiling. And so how reverse proteomic profiling works is you observe for a particular gene in which knockout it gets differentially expressed. Okay, so it's the opposite of a classic proteomic profile. The power is that a reverse proteomic profile includes the information you record from all of the 4,500 proteomes that we have measured. Yeah? So the statistical power increases, and therefore for many genes we find many more responsiveness and interactions with reverse proteomic profile. Another thing we can do, and this was very successful in metabolomics, we can take the proteome just as a profile and cluster it. And the assumption is if in a big matrix, two proteins cluster very closely, they have a more related function. This works, but it turns out it's more challenging in proteomics as it was in metabolomics because the dimensionality of the data set is pretty high. But because of the high dimensionality, we learned we can do something else. We can look at co-regulation patterns. Now we can look at protein A. Let's see some mitochondrial protein, and we can see how this mitochondrial protein changes in all of the knockouts. And then we can look for other proteins which have a similar pattern across the knockouts. And then we can calculate a distance matrix. And it turns out that this type of analysis gives us the best information about protein function that we have seen so far. Okay, this is an example here. This is a precision recall curve from some gold standard databases. And you see that all of the methods nicely recapture the known biological function. But the clear winner in terms of how much information you get are protein co-regulation patterns. So they clearly exceed in reproducing the common knowledge. Okay, now another nice thing of these approaches is that we have an abundance bias in the proteins we can quantify in proteomics, but we also have an abundance bias in the importance of proteins. No? So proteins which tend to be essential genes, important, well connected in the cell, they tend to be high abundant. Well, actually, it's a quite nice correlation. Most of the high abundant proteins are essential ones and highly connected. And those we can never knock out by genetics, because if they knock them out, either we kill the cell, we create a lethal phenotype, or we perturb the cell so strongly that the growth rate goes down and any omic profile is useless. But many of the not so connected proteins, they you can knock out, and many of them are low abundant, which means in the end of the day, it turns out that you're quite complementary and between the genes that we can knock out and the proteins that we can quantify. Yeah? And once we combine the knockouts with the quantified proteins, we come to close to a saturation to what we expect is active in a microbial cell at one given point of time. Okay, so this gives us a lot of information about the proteins. It's about to be published, so not giving you much more here, but I tell you some other things you can find out with a data set like ours. So one of the first one was a paradigm. I always grew up in learning that protein half-life, the turnover rate, and the abundance or the response pattern measured by abundance in a cell are totally something different and not related to one another. It turns out that this is not the case. So this is a very simple machine learning method trained on our proteomic data, and we try to predict protein half-life. And actually what you see here is that protein half-life is pretty nicely predicted from its response pattern in knockouts. Okay, so protein abundance and protein half-life are not independent properties. They are connected to one another. And this is nice because measuring protein abundance is much easier than measuring half-life. And if you now have the abilities to connect the two, you can derive relatively good half-life values from the abundance of a protein. Yeah, and the same turns out for translation efficiency. It's not independent of the abundance of a protein. Once we know the abundance data of all the proteins in the knockouts and we train a simple machine learning predictor, we get pretty nice in predicting that translation efficiency as measured with ribosome profile. Okay, so those big proteomic data sets allow you connect the different biological properties. And another example that I wanted to share is we can now do other things. For instance, we can look at all protein complexes at the same time, because of course we have perturbed by all non-essential genes, means we have perturbed all of the non-essential complex members of every non-essential protein complex. Okay, and this allows you to answer some general questions. So one of the paradigms we wanted to test is this importance of the non-exponential degradation paradigm. So the non-exponential degradation paradigm says that that most protein complexes are made in a way that the central subunit is produced, then the protein complex assembles and destabilizes the proteins, and then the new complex members, which are then translated, they get degraded much quicker. So an implication of the non-exponential degradation hypothesis is that once you remove a protein complex or its core subunit, that the remaining subunits will get degraded as well. And we tested this in our data set, and what you can see is this proves totally true for about 30% of the protein complexes. Yeah, so you delete one subunit and the other ones get degraded. You can see this here on the coaptomer complex as an example here. That's the typical abundance, and once you knock out the key subunits, the remaining subunits get degraded. Okay. Now, there is some 50% of the protein complexes where we cannot say anything because we didn't see anything. That could mean that they're either not responsive in this way, or just that we didn't hit the essential subunit because we cannot knock out essential genes. But it turns out we have some 20% of the protein complexes which defy this paradigm and do the opposite. And one example here is the glycine decarboxylase complex. No? So this is the abundance of the complex members in the wild type strains. And once you knock out the key subunits, you see that the remaining subunits then increase in abundance. So they do the exact opposite what the non-exponential decay hypothesis would have predicted. And then we are thinking about this, and actually it's relatively straightforward to understand. It looks like these are the protein complexes that are regulated by a feedback loop. So if you delete the glycine decarboxylase complex, the glycine concentration increases, that you can see here. So the glycine concentration dramatically increases in the cell. And it's known that glycine levels act on a transcription factor which regulates the glycine decarboxylase complex expression. Yeah? So that means the moment you have more glycine, the cell wants to degrade more glycine and activates the complex. And thus it's not functionally, that becomes in a loop that is not effective. And the same is true for another example here. This is the proteasome. Proteasome is regulated by the amount of misfolded proteins. So the moment you perturb the proteasome, this doesn't work anymore. There's a feedback that's driven by a transcription factor called RPN4. And so once you do this in a wild type cell, you see that upon perturbation of the proteasome, the other complex subunits increases. And if you delete the transcription factor, this process is reversed. Okay. Last but not least, we can now look at function, yeah, and we can actually perturb the different functional categories, because we do this when we delete all of the genes, and then we can say which other processes are connected and enrich. And so this gives you a genome-wide cause and consequence map by whatever process you perturb, which one is the one which responds. And for me as metabolism guy, one thing turned out to be quite exciting. So whatever you do to the cell, the most likely thing to give a response is amino acid metabolism or a nucleotide metabolism. So the cell typically reconfigures to whatever you do to it by changing metabolism. And this is some knowledge we would have had to know 10 years before when we started to design. But of course, there are many more specific processes that you perturb and many specific responses you can have. And so there is something in these maps for everyone. So if you have your favorite process you want to study, look at some of those maps and you can probably find how they connect to others. Okay. And last but not least, I thought I'd give you just one example to see how useful this can be to work out some old paradigms. And there is this very important and profound knowledge that, slow growing cells, they have a different proteome. Yeah. And you can see this here a little bit in this box plot. So this is slow growers and normal growing cells sorted. And then you look at the proteomic profile and you see that the slow growers have much broader proteomic changes. Yeah? And this is known in the yeast field, and always when I present at conferences, I get the feedback and people tell me, oh my God, yeah, you have just found a very expensive method called proteomics to measure growth rates. But these two plots now show that it's more complicated as you think. So the first thing, in this proteome, there is the information imprinted how fast the cells grow. Again, this is a very simple machine learning method, and we try to predict from this proteomic profile here the growth rate. Yeah, and what you actually see is that this predicted growth rate, so the growth rate predicted from the proteome, is pretty much matches the measured growth rate of the cells. Yeah, so a proteomic profile is enough to tell you how fast it grows. Yeah, so that far that clear. But then the question is this really explaining these profiles? And there we had a surprise. So here, these are the proteome profiles of the slow growers, and what we see here in green are the growth-related proteins. Yeah, and if the slow growth would entirely be explained by the differential expression of the growth-related proteins, this would explain the proteomic profile. But we see that actually the majority of differential protein expression in the slow growers is from proteins which are not growth related. Okay, so it's non-growth related proteins which dominate the profile of the slow growers. And this was puzzling initially. We said, okay, what can be the reason? And there are of course many reasons, but we found one key one. And one key one is that we induce chromosomal aneuploidies in the cells. Now, if you delete certain genes, and the cell compensates, and very often the compensation is a chromosomal aneuploidy. That's an example here. If you delete the gene called NAP133, and 2 chromosomes duplicate. Now, this is chromosome 1 and chromosome 3. Another example here is if you delete CRE28, you duplicate chromosome 7 and chromosome 8. Yeah, and you see this pretty nicely on the proteomic profile because the proteins that are located on that chromosome, they get induced. So why this is the case so very often, and these are chromosomes that contain compensatory proteins to the lack of a gene, so that's known from cancer biology. And what we see that this is so nicely presented at the proteome that we can actually nicely set cutoffs and identify a chromosomal aneuploidies from a proteomic data set. Yeah, so this is an example here of all of these chromosomes, and you nicely see those knockouts, for instance, which in this case I think it was chromosome 5 or 6 that is here duplicated. Okay. Okay, so now every gene which sits on this chromosome, of course, will be differentially expressed just for the reason it sits on the chromosome. And this has not necessarily anything to do with the reason why this strain grows. And then we asked if these aneuploidies explain broad proteomic profiles in the slow growers, and that's absolutely the case. So a lot of the broad proteomic profiles in slow growers are caused by the proteins which just sit on the chromosome that is becoming aneuploid because you genetically perturbed the cell. Okay, so in this way, proteomics can help you a lot to identify the proteomic changes. Okay, I see I took me a little bit longer already in time, as I wanted to go, so I skip over the last example. Just one other thing, we can use the same methods that we use for systems biology in medicine. We have done this a lot during the COVID pandemic, as Tianan also has done. Just very briefly, we can accelerate with our methods this significantly. These are some 60-second gradients on human plasma, and they nicely classify COVID patients. They identify proteins which are differentially expressed in COVID patients. And we can use such signatures to predict the outcome of COVID patients. So this is a survival curve from our most severely affected IPS patients. So the proteome is very predictive about the outcome of those patients, and we can actually discriminate, and we can use those things to monitor clinical trials. And because we were able to extract the key peptides from our proteomic data and develop targeted MRM panels for those, and they could be implemented in the clinical laboratory, and our clinicians can now ask for those tests when they see new COVID-19 patients and make a judgment about how they should be treated and how the outcome should be. And very recently, we moved such things also to other disease and tested whether it works in other infectious disease, and we have good success. This is an example here for monkeypox. We find very clear response pattern of monkeypox proteins, and they tell us how severely ill a patient is. And an interesting new possibility came up in this way. We realized that the human host response, the innate immune system response, is so similar between different viral infections that you can actually take a panel assay that has been developed, let's say, for COVID-19, and you apply it on monkeypox disease, and you still get a classification of the patients because there is a significant amount of overlap of the host response proteins. And that means that for future pandemics, you can prepare now panel assays which are agnostic about which viral infection is triggering your innate immune system. And the only thing you need to change in the end of the day is then the machine learning model that gives the outcome prediction. And we hope that things like this are massively accelerating our discoveries. With this, I come to the end of my talk. Thanks for staying with me. So at lunchtime, also early in the morning, also late at night, like in California. This was teamwork of a great team. Just to highlight Christoph, who developed most of the high throughput proteomic methods that I presented to do, and Vadim, who is now a junior group leader, and he was as a postdoc in my lab developing Diane. Yeah, thanks so much for staying with me, and I hope there are some questions that I could answer.
Thanks, Marcus. Very impressive. I like to have a few questions. Maybe if other people have a question, please leave in the chat window. So I'm quite curious. You mentioned you can analyze 1,800 samples per week. It's really reaching the limit. Is this the limit of proteomic analysis? So I wonder what's the depth? Do you worry about the depth, and also how about the stability of the system? And do you have a limitation or bottleneck in the sample preparation instead of the mass spec?
Yeah, well, all of these can become limiting, right? The stability is actually one of the least worries. So this improved a lot by moving to analytical technologies. Now they are just more mature than these nanoflow and microflow systems that we always used in proteomics. If you go to industry style HPLCs from Agilent, for instance, now they are just at a different level of stability. Yeah? And the other thing, the high flow rates, they equilibrate your columns faster and better. There is less carryover. You know, there is just less problem on the chromatography side, less problem with cloaking if a sample is contaminated at this high flow rate. You know, this no dead volume problems anymore. So a lot of the conventional problems that annoy you as a proteomic lab, they are less a problem in high flow rates. You make the instruments more dirty. Because you inject more samples, so that's one new limitation. Yeah, but this is now getting better with the latest generation of mass specs. They are again more sensitive, so we can inject now less sample, and that makes it less dirty. Okay, can we go much faster? At the moment, it doesn't make sense. So we are already at the same speed as you would do in a flow injection, but with the short chromatography, we get a much better quality of the data as with a flow injection. Yeah, so going much faster than a 30-second gradient, I think it will be hard for proteomics, at least for the time being. Let's see what the future brings.
So
yeah, in sample prep you can scale.
How's the lifetime of the column?
Oh, fantastic! So we have some of those high flow columns which do 10,000 injections. Yeah.
Oh, cool. I'm also very curious about your ArcScans mass specs. This is really cool. You show that after you remove the glycan, then the picture becomes completely white. That means you really did a very good enrichment of the glycopeptides or glycans. So have you tried this in deeper screening of the glycopeptides or proteins in the plasma? Because there are so many low abundance proteins which are glyco, and people have found a lot of analytical methods to enrich the glycopeptide for that.
Yeah, but we haven't. Yeah. So our main motivation is actually to come up with a method where you don't need to enrich, because in the moment you enrich, you're already in the scientific setting. No, you need to be at low sample numbers. You have a lot of noise. You can discover new things, and we wanted to have a complement where we can screen, where we can measure hundreds or thousands of patient samples. No, and so Oxoscan MS is designed as a screening method. It's meant here to measure a cohort or to measure a systems biology screen. And then many of the signals we see, you still need then to deep dive then, because the oxonium signature alone is not perfectly identifying the peptide, and it tells you that's the retention time, that's the exact mass, there is a glycan on the peptide. Yeah, and what you typically then use this information, then you go to a high resolution mass spec with a long gradient in the next step, and then you perfectly verify the structure that is predicted or you assign it. We are working on a new method at the moment which would do this in one go, to sequence the glycan, to sequence the peptide, and to quantify it in one rush. But at the moment, this is still a two-step procedure. Now, the Oxoscan, it's more like a metabolomics workflow than a proteomics workflow. The Oxoscan quantifies and tells you where it is, but it's not uniquely identifying it just yet. And that's why you then need to go still with the classic method then to assign all the sequences.
Did you run in a DDA mode or DIA mode?
So Oxoscan is a DIA method, yeah, but the validation we did with a DDA method then, yeah. And so the validation.
Yeah, maybe a library and then using dye to match those features. Do you have such a library?
Yeah, we're building one. Yeah, we use public libraries from other people who make good libraries, and they then seems to work well. So this is still work very much early stage, but it's a cool method, and you should try it out, and hopefully you're gonna like it.
Good. Is there any other questions from the audience? If not, thanks a lot, Marcus. Let's move to the.
Thanks, everyone. Hey, Jurek, everyone else. Have a good morning.
Thank
Ceci.
you, Marcus. Yeah. Our next lightning talk is a recorded video. I will share that. So give me 1 second, please. So lightning talk 3 is the autonomic approach towards a better understanding of chronic lung inflammation. Pierre Haggis from the Leeds University, Belgium. Unfortunately, he won't be able to present, so I sent the recording. Let me play with that. Sorry.
Today to discuss about our research on volatile-omics approach towards a better understanding of chronic lung inflammation. One of the most famous chronic lung inflammation is asthma. And asthma is a major concern because its burden is increasing over the years. This is mainly due to the degradation of air quality and the increase in most of the country of air pollution. We estimate that more than 300 million people are suffering from asthma, and a high percentage of those people are children. The problem is we cannot cure asthma. We can treat the symptom, and treating those symptoms is really expensive. We spend almost 20 billion euros in Europe per year to treat asthma symptoms. And it's crazy to think that 20% of the patients are receiving the wrong medication. So to improve our way to monitor asthma, we need to understand the different type of asthma. So in asthma, you have chronic inflammation, but the sources of this inflammation can vary from one person to another. So we have inflammatory phenotype due to the type of inflammatory cells that are recruiting inside the lung. So to try to differentiate a patient with different phenotype of inflammation, we conducted a large study between Maastricht University and Liège University in Belgium, where we analyzed the exhaled breaths of patients trying to better phenotype asthmatic patients. So we recruited more than 500 people in 2 phases. So the first phase was a discovery study in Maastricht University, where they screened the breaths of half of the patient. They identified 7 potential biomarkers, and we validate those biomarkers on a second study with another technique in another lab. So to analyze the breath of those patients, so this small molecule, we use gas chromatography, but not any type of gas chromatography. We use multidimensional gas chromatography coupled to high-resolution mass spectrometry. And this is the typical chromatogram that you obtain. In multidimensional chromatography, you have a first separation on a GC column that is similar to 1D GC, but we also add an extra separation dimension, a short GC column, to create a second dimension, and we obtain this 2D chromatogram that are an image, a fingerprint of the sample composition with a structure separation. That means that the location of the peak gives us a hint about the identity of those peaks. And based on those hints, you can add electron ionization, where you have the fragmentogram that you can compare with a library like the NIST of Wiley. In addition of that, we have high resolution, so we can have molecular mass simulation with less than 2 ppm accuracy. And putting all of that together, we have multi-factor identification, where we have fragmentogram, high resolution, GC linear retention indices, elution pattern in GCTC. So for those really small molecules that are the volatile compounds, you can reach level 2 MSI identification even without purchasing analytical standard. So it makes your life much more easier because the putative list that you have to validate, it's already like a step closer to validation. So based on those compounds that we validated, we compare what VOC can do for identifying eosinophilic asthma compared to what is used in clinic, exhaled nitric oxide and blood cell counts. And you can see that we obtained the same figure of merits than those clinical methods. But what we did is in terms of data processing, we also combined those three screening methods, and we obtained the green rock on the left, which is much better than the three individual ones. So it's really interesting to see that we are not changing completely clinical practices. We are just providing an extra tool to have extra confirmation of asthma phenotype. So this is the 5 markers that we use for our VOC classification model. For those ones, we purchased the standard and we did multi-technique confirmation. So we know that those compounds were the ones that we detect in the breath of the patient. So now the question is, okay, you have those compounds. So what next? So now, like from this study, we have multiple studies that are like coming down the line. It's how can we make these techniques a routine practice in clinic? Because providing a GC/GC IRS to clinician, it's really complicated and really expensive. So we have to shift to something that is more like transferable to clinic. And also, we need to understand where these compounds are coming from and what is the impact that they have on the human metabolism. And is this part of the research that I want to discuss with you today. And our best guess, if you look to the literature, is that those compounds are coming from oxidative stress. And we know from previous studies that oxidative stress is coming from a lot of different factors. And what is generating? It's generating lipids peroxidation, and this byproduct of the peroxidation is VOC production. So how can we model this oxidative stress and understand it at the molecular level? So we took a lung epithelial cell line that's commercially available, A549, and what we did is we stress it with oxidation agent, a chemical stress with H2O2, and also a biological stress using sputum from asthmatic patients. So taking an ex vivo fluid coming from the lung of asthma patient. And we spend a lot of time making sure that the VOC and the metabolic perturbation that we create is really coming from the oxidation. So we designed this study focusing on controls. And we wanted to have biological control, so the impact of the biology, the impact of the technique with analytical control, and physicochemical control, because we have 2 phases, so we need to make sure that it's not like perturbation just due to that change in the equilibrium phase between liquid and gas. And the first PCA that we obtained, we can clearly see like where we have cell, where we have the control without cell, so we are going in the good direction. So first, we optimized the H2O2 condition because we wanted to generate oxidation, but we didn't want to kill the cell. So we tried different exposition time with different concentration. And at the end, we selected 60-minute exposition time with 0.1 millimolar, where we generate oxidation without killing the cell, which can be seen on the PCA. So in green, you see the cells that are untreated, just diluted with a bit of PBS. And after we have the blue cell with long exposition, high concentration when we have cell death, so apoptosis. And the purple and the red are the 2 oxidation conditions. So based on the concentration, you can see that you are going down PC2, so you are generating more and more oxidation, but the cells are still alive. So we perturbate the metabolism, but we didn't kill the cell. So this is what we wanted to obtain. Next, we move to the biological stress, where we pull sputum from asthmatic patient. And what we obtain after 24 hours exposition time, it's cell proliferation. And cell proliferation, it's exactly what you see in the lung of patients with chronic inflammation. So we were able to mimic what's happening in the alveolar level in the lung by having this cell proliferation due to inflammation. And taking a pool of healthy control, so the sputum from people without chronic lung inflammation, nothing happened at the same concentration. So really the inflammation mediator present in the sputum are generating its cellular proliferation. So that was really interesting to have this control too on the biological side. So at the end, we obtained a list of compounds that have putative identification. And even if we didn't validate yet all those compounds, what was interesting is to see that we found our usual suspects. So when we analyze the breath of a patient with chronic inflammation, like a small chain aldehyde, branch hydrocarbons, are compounds that we usually detect. And they were also generating in vitro, showing that our in vitro model is on the way of mimicking what's happening in the lung, even if there's still work to do. So take-home message, the future of exhaled breath requires more fundamental understanding. So screening breath of patients like thousands of people did for COVID, it's nice, but you need to understand what's happening because there is so many bias on the analytical and biological side that you need to make sure that you design it properly. In vitro inflammation modeling, it's a key player to implement multi-omics workflow. So if we want to do multi-omics to understand where these compounds are coming from, we need in vitro model to generate enough sample. Volatile metabolomics or volatolomics as a seat at the table of multi-omics. So I think there is a growing interest for this small molecule screening, and I think that it's really part of the multi-omics family. And as I mentioned it several times, a robust design will always be the key for meaningful information. So make sure that you design first before to run the analysis. If you want to know more about the technique that we use for this study, I invite you to a workshop in Liège in January 2023. The event is completely free, so don't hesitate to register, join us, and have fun discovering more about multidimensional chromatography. And I want to thank you for your attention.
Thank you very much. But unfortunately, if you have any question, please send that to me. I will forward that to him. So we'll connect you with him to get more information. So our next speaker, lightning speaker, Tianan can introduce. Let me share the screen. Tianan, do you want to introduce? So,
sorry, Susie, I think Tiana is not available at this moment. So maybe you
okay.
can introduce the
Yeah,
speaker.
I will do that.
Thank you.
Yeah. It's my great pleasure to introduce Amit Yadav. I hope I pronounce your name correctly, Amit. You're going to present, you're from Translation Health Science and Technology Institute, India. And you're going to talk about large-scale analysis of protein post-translation modification, PTMs, from the human proteome and mass spectrometry data. Floor is yours. I'll stop sharing.
Thank you. Am I audible? Can you see my slides?
Yeah, that's better. That's better.
Okay, thank you. Thank you for the introduction. And thank you for VPAP 2022 organizers for having me here. I'm going to talk about protein post-translational modifications, how we can have a large-scale analysis, and also looking at mass spectrometry data to identify and localize different post-translational modifications. So whenever I begin, I show this slide to tell that when we talk about the different types of omics, genomics is probably more closer to the blueprint, while with the effect of diet and environment, we have changes which are specific to a condition, which we generally see in transcriptomics data, which tells you what is likely to happen. And proteomics and metabolomics are probably more closer to the phenotype, which tells us what is happening with the virtue of the enzymes and all other kinds of structural and functional proteins. And metabolomics tells us what has happened. So in collaboration with proteins and metabolome, we can actually deduce the phenotype, which is also kind of shown by Dr. Ulcer earlier. He told us that gene knockouts can be more closely predicted by translatome data and metabolome data. So that is also the kind of underlying message here. So it means that we need to identify proteins in more detail. I look at proteins more than metabolome because proteins are a versatile kind of biomolecules that talk to DNA, that talk to RNA. They also talk to themselves in the form of protein-protein interactions, and they also talk to metabolites. So we see that different kind of proteoforms or mod forms can be formed from a limited number of genes, which provides us this diversity of complex phenotypes that we can have in health and disease. So it is quite important that when we measure proteins, we just do not stay at the level of talking about these are the number of proteins that we have identified. We should also look at in more detail and find out whether they are occurring in one or multiple different kind of proteoforms, many of which are caused by protein post-translational modifications. This is the recent perspective in NCP that talks in detail about this concept. And while I was working on liver diseases, we checked that almost 90% of liver diseases have one or more PTM sites. And when we talk about the whole human proteome, we see that roughly on all the chromosomes, on an average, 70% of the proteins are modified. So when we talk about specific set of PTM sites occurring in a disease condition, it tells us a lot about how we target a disease, develop the targets or biomarkers. But if we just look from above and look at the forest rather than the trees, we wish to identify if there are some emergent properties of the human so-called PTMome. The dark matter of biology has been used in different contexts for different people, and for people like me who are doing proteomics, we tend to refer this term to post-translational modification. So just to have an understanding at large scale, we downloaded data from Nextprot and classified it to different categories. We wanted to see PTM disease associations, whether how PTM functional diversity imparts its properties to the proteome as well, whether PTM disorder has some association. I will be leaving this part out today because I don't have that much time. Then how PTM then variants are associated. So on analysis, we found that when we compare the proteins that are known to be associated with some disease versus the ones that are not associated with the disease, the majority of changes that we see are in the top. Maybe you are not able to see my. Okay, so the bottom blue part, light blue, and the top dark blue part are the ones that actually change. And if you look at the sum of those, it kind of says that the other categories where 1, 2, 3, and 4, 5 PTM sites are there, they remain roughly the same. But in the disease condition, we have multiple PTM site fraction to be increasing because the number of proteins are not the same. That's why we are looking at percentages, because it gives us a more balanced perspective. So we get a very significant p-value, and the idea that we understood from this was that multiply modified proteins have a very strong association with different kinds of human diseases. We also wanted to look whether this is important in context of normal diseases, or is it something peculiar with housekeeping proteins. So we find out also mentioned by Professor Ralser also that if we look at the bottom blue part, it says that the housekeeping proteins cannot tolerate too much of disease condition. If we talk about increasing number of PTM cells, it could be because they might be lethal. But if you look at the green part here, as the number of PTMs increases, the frequency of disease in the non-housekeeping proteins keeps increasing. So it tells us that multiply modified proteins, which are not really housekeeping proteins, they are more prone to be involved in disease. So when we were talking about, we are trying to understand functional diversity of the whole proteome, how does multiple PTM protein and disease susceptibility connect to this? We found out that if the number of isoforms of a given protein increase and the number of PTMs increase, and the circle size represents number of protein-protein interactions here. We only took the proteins that have more than five or more biological functions, and we found out that it's a complex interplay of either the number of PTM sites or the number of isoforms, or if both of them are quite less, then in that case the number of protein-protein interactions are high, and they were because of either PTMs or because of other surface accessibility areas that could help them to interact with a diversity of proteins. We also looked at that when the PTM frequency increases and we have only a high function proteins with high number of biological functions, we see that most of the proteins that are involved in disease, shown here in red, are on the top panel. So probably this tells us that PTMs have some strong association with disease. We found out that most of the cancer-related proteins and transcription factors that are master regulators are found in this region. So we found out that the functional diversity of these multiple PTM containing proteins have a very strong association with disease susceptibility. While these ideas look great, what could be the mechanistic basis? So this is kind of documented in many papers that loss or gain of PTMs actually help you gain or lose PTM sites that may be involved in some specific signaling. There are several examples known for this. So we thought, what if we take disease and SNP associations and try to see whether they are mapping to a PTM site loss, because site loss is much easier to study than gain of function. So we took the cosmic and BB-SNP databases, filtered out the synonymous SNPs, and then within a plus minus 7 amino acid stretch around a PTM site, we tried to see whether they map overlap or not. We found that roughly 40% of these could be directly mapped to PTM disruptions causing the disease. And in this figure, we can easily see that phosphorylation, ubiquitination, and acetylations are the most studied ones. So this could be a bias of the studies doing this. So we wanted to ask if we could actually apply this to a disease condition per se, and not to the whole human proteome, whether we can use it to study a particular disease. So we took an example of certain interactors. Certains are important proteins that are known to help in cardiovascular diseases. So what we did, we took this interaction networks from 5 different interaction databases, and we used 3 different PTM databases to fish out all the important proteins that have these post-translational modifications. We have roughly around 100,000 PTM sites, and we mapped all these PTM sites in the certain interactors and tried to study their crosstalk. So what is a crosstalk and hotspot? So if the green site is the site I'm trying to study, site of PTM, and in a plus minus 7 amino acid residue, I see more PTM sites in the same region. This we termed as a PTM hotspot. That means this region tries to have as many PTMs as possible. In the case of crosstalk, if a site is competing for another PTM, and again in a plus minus 7 amino acid residue, there are other such competing sites. We call, if there are more than 3 such competing sites in the same region, we call them as PTM crosstalk hotspots. So it is understood and well documented that different kind of such hotspots are more involved in disease. And that's what we also found when we tried to look at the number of proteins that could occur in hotspot and crosstalk genes. We found that more than 50% of our proteins had either 10 or more crosstalk or PTM hotspot. In terms of cardiovascular diseases, we found that roughly 50% of the PTM hotspots are associated with disease, while a similar ratio was there for other diseases. But more than that was involved in PTM crosstalk hotspots. That means PTM crosstalks might play a more important role in cardiovascular diseases. So we also looked at disease variants and tried to see that a simple polymorphism, disease-associated polymorphism, or the ones that are not yet classified into either, whether they occur with the in situ crosstalk. In situ crosstalk means when the 2 PTMs are competing against each other. And no in situ means they may be in the vicinity. So we see whether we talk about in situ or not in situ crosstalk hotspots. We see that in CVDs, the proportion is always high as compared to the other. And this proportion closely matches the ones that are not yet classified. This means that it is possible that many of these may be actually also involved in disease, but their function is yet to be determined. So we also found that let us fish out the ones that are multiply associated with such variants and have a lot of PTM hotspot and crosstalks. And we found some list of important proteins. So lamin A is the one we left out TP53 because the role is well known in cancers. Also, it is quite an important regulator of cardiac homeostasis and embryogenesis. But we left it out because a lot of studies were there on this. And lamin A is the one that with my collaborator at NIPER, we are trying to study in more detail. There are other proteins that regulate either metabolic processes or response to oxidative stress, and they may be important for other metabolic diseases as well, but that remains to be determined. So this all looks fine on large scale data that others have generated. So we wanted to find out if we can also develop tools and try to find this dark matter. There are several challenges with this, although there are several tools also. So MODE is one of the fastest tools available, but we find that there are a lot of false positives in identification, site localization, but there is no solution for PTM combinations. Towards this goal, we started developing certain tools. So I'm skipping this part because we simply try to parallelize the given tool so that we can identify things faster. Mod locator is an algorithm that has been devised from one of my previous works on masses, where we looked at intensity of the ions as well as the continuity of the ions so that we can determine the location of a site correctly. How is it different from A-score and other site localization algorithms is the important key of intensity as well as continuity of ions. Others are statistically based and give quite good results, but we have seen on some standard data sets where we tested against methionine oxidation, we found out that we could correctly localize many of the modifications and also find newer amino acid specificities if we match with the Unimod database. So we could see that there were certain errors that we could fix. We developed PTM annotator because all of these algorithms give us a mass that we need to manually annotate using Unimod or any other such PTM database. So we simulated a database of a data set of 3.7 million simulated spectra using 4 different PTM and their combinations for standard 18 proteins. So although we know that there are certain isobaric masses because of which there could be certain errors in this annotation, but we could correctly annotate others. So mostly 97 to 98% of the time it works correctly. And the only errors that we see are because of isobaric masses. And if you look at amino acid propensities for a different kind of modifications, we are likely to solve these. So this is another example of PTM annotation. So we look at, so we take data from Unimod and we tell also the class of PTM that we have found out, whether it is a chemical artifact or it is a post-translational registration. So for these different mod form identifications, we can either use label-based or label-free methods. So I'm just giving one example here because I'm running out of time. So we can use a spectral counting-based method and looking at the spectra that have been used to identify either modified or non-modified peptides, we can define the fraction of proteins or the mod forms that are in different states. So since this was only a dummy, small 5,000 spectra example, this was just under pilot testing. I'm not showing different kind of mod forms, only mod and non-mod. Otherwise, in a real biological data set, we may have different forms for a single protein given depending on the type of PTM sites that we have identified. The important take-home message here is that there are certain proteins that are only present in modified form, which kind of breaks the computational tools axiom, which believes that for the proteins that are found in non-modified state, we generally dig deeper and look at their modified form. So it's not always true. So thank you. With this, I come to the end of the talk. We have also applied these tools to non-alcoholic fatty liver disease, and maybe this is this will be shared on some other forum where I have more time. Thank you for the organizers. Thank you, Dr. Bishopri Mishra and Priti and Janan Guo for arranging this wonderful work, wonderful online forum where I got to talk about this. And I also thank my lab members, also Ruchi and Pallavi, who did major work that I have shown here. Some of this is unpublished. Only one paper on PTL crosstalk was out there. We have lots of interesting results, which we will see in due time. Thank you.
Thank you, Amit. Really nice talk. There's one question I can see in the chat. Salia, it's a great work. Was just curious to know. So does the pipeline also differentiate between oxidation as artifact and as positive modification?
Yes, it can. Since we are using Unimod data, this was just an example where we knowingly put oxidation. This oxidation that I was showing here was a chemical artifact. And if it is biological modification, and if we are able to localize it on amino acids other than, let's say, methionine, histidine, or tryptophan, which are known to be modified because of your carbonyl methylation. So in such a scenario, we would be able to differentiate between biological ones and chemical artifacts. Although there are other better methods for doing so, but on a high throughput scale, I think this is the best one.
Yeah. Thank you, Amit. Biswa, you have a question?
Hey, Tusi, thank you. Amit, am I audible? The question is, whenever you do these open searches, what are the best ways of validating some of those finds? Is there any orthogonal techniques that you would recommend?
So, I mean, it's an interesting question. I would not say that I'm the best expert on this, but usually unless I'm using some orthogonal technique, I would rather go with the first step as being the manual validation of spectra. That is the first and foremost step. But for orthogonal technique, you can definitely go for some targeted proteomic studies or synthetic peptides. That will be best. But I would suggest do that only when you have identified something that you wish to be you wish to take further for biological studies as a biomarker, or you have identified it quite significantly among different biological states that you are using. Otherwise, it would be not recommended.
Got it, Amit. Thank you.
Targeted remains the best. Targeted remains the best method.
Thank you. So solutions for more proteomics is even more proteomics. Thank you.
Because of the time, we better move on. Thank you very much, Amit. And if you see any questions popping up, please feel free to answer.
Sure. Thank you.
We share our last lightning talk speaker, Dana Hicks. Dana from Australian National Phenomics Centre in Perth, Australia. Dana is going to present optimization of a tissue homogenization and extraction workflow for comprehensive lipid profiling. Floor is yours, Dana. Sorry for the delay, and I'll stop sharing.
That's all right. Thank you. I'll just share my screen. It was. Now it's disappearing. Share. Yeah. Has that got it?
Yeah, perfect.
Oh, thank you. Hi, everyone. My name's Dana, and I'm actually a master's student at the Australian National Phenome Centre in Perth. And my project was a lipidomic-based project where we were actually looking at optimising lipid extraction from biological tissues. So lipidomics enables the identification and quantification of the lipidome in many complex cellular processes, which are attributed to metabolic diseases. Many of these processes occur in tissues such as the liver, heart, and adipose tissue. So analysis of these tissues provides us with valuable information, especially in model organisms such as mice. Recent advancements in ultra high performance liquid chromatography, tandem mass spectrometry, electrospray ionization, and multiple reaction monitoring provide us the optimal sensitivity and specificity required for comprehensive and robust analysis of the lipids in these tissues. Sample preparation for these is often undertaken by liquid-to-liquid extraction, which enables the solubilization of the lipids from the tissues into the extraction solvent. When I was actually starting this project, I was actually quite surprised when I was looking at literature that there tends to be a blanket LLE approach that's commonly used in lipidomic studies, where they actually use multiple different tissue types, even though they each have quite distinct complex structure and properties. So there's currently numerous methods used to extract lipids from tissues. However, a comprehensive analysis of the efficiency and reproducibility of these methods to extract multiple lipid classes across different tissue types has not yet been performed. So I chose 5 extraction methods from the literature to test in murine, adipose, liver, and heart tissue. To effectively assess the reproducibility, 6 technical replicates for each extraction method across each tissue type were used. However, owing to the limited amount of tissue available from each mouse, this actually needed to be pulled from several animals from the same cohort. So I actually used freeze-dried tissues, and that actually enabled to remove the biological variability amongst those tissues as well. So I used PCA and coefficient of variation to assess the efficiency and reproducibility from these. So several pilot tests were conducted to assess the ideal homogenization speeds and the sample concentrations, and we use targeted analysis with Science QTrap 6500. Now, the benefit of that is that we have the multiple reaction monitoring and electrospray ionization, and we use 71 internal standards, which enabled the relative quantification of 20 lipid subclasses. So this schematic provides an overview of the first development phase, where we actually use the freeze-dried tissue. Subsequently, we ended up actually using biopsy tissue because that's more of a representation of real-world studies. So these were actually, the freeze-dried tissue was actually in liver tissue, heart tissue, and adipose tissue. And each of the extraction solvents I selected have different varying levels of polarity and dielectric constants. So the IPA 90, that was the most polar extraction solvent. So that's actually a mixture of IPA with 10% water. I then had a look at BUME 1:1 and BUME 3:1. BUME 1:1 tends to be in the literature more so for plasma type work, whereas BUME 3:1 is being used in tissues. Then I'd used 2 different MTBE extractions, and the only difference is the second one I actually added ammonium acetate because I wanted to actually see how that had an effect on the actual reproducibility. And then chose the dichloromethane and methanol extraction 3:1 because that's quite often used as a chloroform substitute in literature. So the results showed that each of the extraction methods was actually quite variable amongst each of the tissue types, which is what I actually suspected. So in the freeze-dried adipose tissue, the MTBE extraction was actually proved to be, something's going on there, the most reproducible. So overall, we actually extracted 975 lipid classes, but what I've actually got listed here is anything with a CV under 30%. And the CV actually that's listed here is actually the median of each of these these lipid species. So as I said, freeze-dried adipose tissue, the MTBE with ammonium acetate was quite significantly different to just MTBE on its own, which was really quite interesting. And we noticed that amongst each of the tissues. The IPA 90 seemed to be quite good overall in each of the freeze-dried tissues. but adipose tissue, it was the BUME 3:1. In the freeze-dried liver tissue, it was the MTB with ammonium acetate. And in freeze-dried heart tissue, it was actually the IPA 90. So each of these that were highlighted in red, we then moved to what I call the method validation phase. So this was actually in biopsy tissue. And so we obviously used, as I said, from the one before, for everything that was highlighted in red that extracted the best. And the results were really quite interesting. So again, we extracted over 900 lipid species with our method. And in the adipose tissue, in the 3:1 for the BUME, it actually extracted the most. It was 889 species. And that was actually quite significant because the only other results I could find amongst the literature was a lipid recovery of 276 lipids from 20 subclasses. In the biopsy liver tissue, the MTB with ammonium acetate was confirmed to be the ideal lipid extraction method with 702 lipid classes, and that surpassed previous reports of 405 lipid species that were covered previously in the literature. Biopsy heart tissue, in this one, it was actually BUME to 1:1, whereas in the freeze-dried tissue, it was the IPA 90. But I actually noticed that we actually need to somewhat improve the reproducibility anyway in heart tissue. And that could literally be a longer homogenization cycle, or perhaps even including freeze drying as an actual pre-treatment step. But overall, the results have shown from this that each tissue type has a distinctive lipid extraction method with respect to coverage and reproducibility of the lipidome. The results also revealed a wide variation in extraction efficiencies from each method for each tissue type. And it hints at essentially that the recovery of specific lipid subclasses isn't solely attributed to how organic or polar an extraction solvent is, but rather inclusive of the actual varying physiochemical properties of each tissue subclass and tissue type present. Furthermore, these robust and comprehensive methods that we will present in a publication that's not done yet have confirmed that the optimal lipid extraction methods are distinctive to each tissue type. And by using them, it'll enable the accurate interpretation of lipid profiles for more meaningful biological analysis. Thank you.
Thank you very much, Dana. Very nice presentation. Is there any question for Dana?
I have a question.
Yes, please go ahead.
Jurek Adamski here, very nice talk. Could you please tell me which of the methods you use for the extraction was working the best with the oxylipins?
With the oxalipids, I don't actually have
ceramides or eicosanoids. You said only classes, but which was working with this oxalipins?
Oh, so they were all quite different in regards to the actual tissue types. But one of the things that I did notice is that the ceramides weren't actually very reproducible at all with any of the methods that we looked at. So that's obviously something that needs to be looked at. They're obviously present, but once we actually filter down to anything across the technical replicates that had a CV below 30%, there was actually very few ceramides at all.
So I would suggest that in your further studies, you will compare the effect of lyophilization, yes or no. So I compare the without and with lyophilization. This is actually introducing a lot of noise in low concentrated molecules among lipids, triacylglycerol, deacylglycerol, ceramides, and so on. So very nice contribution, but it's It's the beginning of the journey.
Yeah, we actually found that by freeze drying, it's a quite good starting platform, because obviously when you're limited to small amounts of tissue, but it really does need to be validated in biopsy tissue. You can't just run it with the freeze dried tissue and then just accept those results.
There's one more question from Sarah Hancock. Great talk, Dana. Sorry, I may have missed it, but are you detecting your lipids in positive or negative ionization mode?
So we use the site 6500, and that actually does positive. Our method actually does the positive and negative mode. It does the polarity switching.
Thank you, Dana. So
thank you.
Yeah, great.
I'm obviously very new to all this, only been a master. Thank you for the opportunity.
Yeah, thank you very much. Great talk. And so move on to our keynote speaker. Tiana, do you wanna introduce? So
I think he will come back into a Wilson goal.
okay, I'll
So
do
yeah.
yeah.
So would you please, thank you.
Yeah. Okay, so our keynote speakers, apologies for running late. Dr. Quan Wang, he's from Shanghai Tech University, China. Dr. Wang is a leading researcher in the field of medical image analysis. His research dedicated to fuse machine learning and artificial intelligence into medical image computing and informatics for the sake of smart diagnosis and prognosis, individualizing therapy planning and tracking, and translational medical studies. Without delay, I'll introduce Dr. Quan Wang. Thank you so much for accepting the invitation, and your title is Deep Learning for Batch Effect Removal and Classification of Metabolomics. Floor is yours. I'll stop sharing.
Yeah, thank you. Thank you for inviting me here and for introducing me. Let me just, which screen? This one. Yes, I think it's the right screen, right? Because I have several screens. Okay. Yeah, thank you. Thank you again for inviting me here. This is Chen, not Ken. Q pronounces like CH in English. It's Chen Wang from Shanghai Tech University. And my title today is about deep learning for batch effect removal and classification of metabolomics.
So sorry.
So, yeah, it's fine. Yeah. So let me introduce myself a bit. I would like to tag me as a medical imaging person. So my major research interest actually covers machine learning in medical imaging. And in recent years, particularly, we are focusing on deep learning technique and its abundant applications in medical imaging and informatics. So to me, I have done a lot of works in image reconstruction, including PET and MR. And we have also done a lot of post-processing researches, such as super resolution and enhancement, as well as 3D rendering of this medical imaging data. And based on this process data, we just do pastillation or try to identify those important regions in brain, in different organs. And we do registration or alignment to compensate for this kind of variability across different subjects. And of course, we do a lot of understanding toward the content of the image, as well as the scene that we have acquired this kind of imaging data. And finally, very importantly, we do a lot of researches on computer-assisted diagnosis, try to develop this kind of, for example, deep learning-based models and apply these kind of models to real clinical scenarios. I can show you, I can just briefly introduce three of my recent works and to help you better understand my research coverage. The first one is about, it's laser. The first one is about the bone age assessment, and we have developed a quite sophisticated deep learning network. Just input this kind of a X-ray image into this network. And plus, we have some gender information and some other lab test, blood test information. Put all these kind of stuffs in and combine them in a network. and this kind of a prediction upon nature assessment could be very accurate. And very important, we have deployed this system to Shanghai Children's Hospital, and we have validated over 100,000, I think, kids in last 3 years. And the second one is about how to read the lung X-ray image. Basically, you can do this kind of a computer-assisted diagnosis or automatic reading to tell whether this patient is suffering from pneumonia or this patient may carry a very small lung nodule. And we have developed this kind of a system. And this system is successfully extended to support the early screening of COVID-19. And we have actually delivered service to more than 200 hospitals in China and in Southeast Asia. And the last one is some of my very recent progress, and I personally very interested in it. So when a radiologist reads this kind of X-ray image, it's about the knee joint, an X-ray image of the knee joint. So basically, when a radiologist tries to read this image, the gaze of this doctor will be recorded by using a gaze tracker, and just stick this gaze track to the bottom of this monitor. And when this radiologist when he or she is doing this kind of diagnosis, we try to record the gaze, the movement of the eyes. So then we can combine this kind of a gaze information, the eye movement information, with the diagnosis report, and then develop a computer-assisted diagnosis system without using any additional man-made labeling. Because if we are doing machine learning, doing deep learning, everyone knows that we have to do a lot of labeling, and that is sometimes very expensive to collect, and a lot of physicians, they actually hate to do that because it's kind of time consuming. So without using this kind of additional labeling, we can build up this kind of a computer-assisted diagnosis system, and that's very fascinating, and we are moving on this direction. So today's topic for me is about deep learning of batch effect removal and classification of metabolic mix. And this is basically based on one of my recent papers in analytic chemistry. And this work is done in collaboration with my friend, Dr. Chen Kun, a professor in Shanghai Jiaotong University. So the motivation of this work is that we know we have a lot of omics data, right? We have heard of many talks today, and we have been talking about the genomics, this kind of data from mRNA or protein, and we have metabolomics. And of course, in medical imaging data, we have like radiomics, which is slightly different from this previous omics data, but they share a lot of things in common. And my lab has done a lot of works on radiomics researches, and we have accumulated a lot of experiences. And this kind of experiences allow us to shift our previous work to, for example, to analyze this metabolomics. And we have we'll see how this kind of technique would be demonstrated. And if we are talking about this kind of omics data, and a very troubling issue is that we have batch effect, right? Because if we are using different device, and if we process this kind of a sample on different days, naturally there's a batch effect. And theoretically, we have no way to avoid this kind of a batch effect. And at the same time, as we have seen in last 10 years, since 2012, in last 10 years, we have seen the very significant progress of deep learning in almost every field, especially in computer vision and in natural language processing. We can just actually input a sequence of sentences or input a volume of images to a network, and this network will do a lot of understanding, processing to help you get the result you want. And this kind of a deep learning has provide a lot of ways to analyze the data and to attain different applications. So our work is actually to show that deep learning can be possibly applied to as a compensation to the batch effect, which I'll show in the next slides. So a basic let's just consider a basic question. Usually, if you are presented with some data in a paradigm of machine learning or deep learning, we can train a classifier or a classification model, and this model will do will finish the classification task. So basically, you have training data, you do this kind of training, and then with the new test data, we do the inference, and this test data could be unseen with respect to the training process. And this kind of a training procedure, we call it a fully supervised training, right? So, but the problem is that often we have the problem to generalize this kind of a classification network from a training data to the new tested data, especially when this training data and test data are coming from different batches, because they have this batch effect, and the batch effect has made the two batches of data, and they have like different distributions. And the implication is that if the two batches of data, they have different distributions, then the model, the network trained from the first batch would, possibly cannot be generalized to a second batch. So if the two data sets, they share like similar distributions, then we will be able to do this kind of generalization. And this kind of argument actually inspires us that whether if we can do this kind of projection, for example, by using deep learning, and if those two data sets, they are they have very different distributions in the very beginning, and after this kind of a proper projection, we can somehow harmonize their distribution, then we can do this kind of a generalization. So based on this assumption, we have designed such a network, and this is the overview of the network architecture. So basically, you can think about it X1 as a sample from batch 1 and X2 as a sample from batch 2. And this network has actually 4 sub-networks. We have a C network, which is for calibrator. Then we have 2 reconstructors, R1 and R2. And then we have a discriminator. Each of these 4 networks actually is very quite straightforward if you are viewing, if you try to understand it from the perspective of deep learning. Basically, this network is actually very lightweight and tiny networks there. So when X1 and X2 are input to a calibrator, this calibrator is designed to make sure Z1, which is projected from X1, and Z2 projected from X2. This Z1 and Z2 would be, share some similar distributions, and that answers the previous the question in the previous slide. If the 2 batches, they have like different distributions, we cannot do this kind of a generalization. But if they have like similar, distributions, we can do this kind of a generalization. So we designed this kind of a calibrator to make sure that even though X1 and X2, they're coming from 2 different batches, and this kind of a batch effect causes a lot of distribution incoherence there, we can use this kind of a calibrator to harmonize their difference and make sure Z1 and Z2 are distributing in a similar way. So this kind of similar distribution is actually enforced by a loss called LC here. LC encourages this Z1 and Z2, to distribute compactly and similarly. So and then this kind of a mapping is actually from a mathematical point of view is called a year post problem, because let's say if you just map X1 to 0 and X2 to 0, and then you have 2 zeros, and these 2 zeros would be very similar with each other. But it's just meaningless, right? Because in this kind of a mapping, you have lost all the useful information. So to avoid such kind of a, we call it mode collapse, or this kind of a collapsing in training, we actually have 2 reconstructors, R1 and R2. Make sure that we can recover X1 from Z1. So basically we have Z1 mapped by R1 and we get X1 prime, and want to make sure X1 prime is very similar, is as close to X1 as possible. In this way, we have some additional constraint on Z1 to encourage Z1 to try to preserve as much information as possible during this kind of a projection from X1 to Z1 and then to X1 prime. And similarly for X2, we have X2, Z2, and passing by R2, we get X2 prime. And we want to make sure X2 prime is very similar with X2. And at the same time, the scenario is that for X1, we know the classification labels, for example, the diagnosis label, the disease labels. So the goal, just remember that the goal is that we can train a model based on the, the data and the label, the disease labels of batch 1, and we want to try to generalize this model to the data in batch 2. So for Z1, for many Z1, we actually can train a discriminator, and this D network will try to finish the diagnosis process based on Z1. For example, just input Z1 to this D network, and you will get a label there. So basically, you can during the training process, you can actually compute the loss of this predicted label in comparison to the ground truth label. And for X2, which is drawing from like batch 2, we don't have this ground truth label, right? Because we're going to do this kind of prediction for the second batch. So that is how data flow happens in training process. And in test process, in inference process, basically the task is that we want to, for each X2, we want to predict its label, its disease label Y2 prime. So in test process, we only need to run the calibrator for X2, and then we get Z2, and we just input this Z2 to the D network, and we get Y2 prime. Remember that this kind of a D network is actually trained based on Z1, which is a mapped code, a mapped projection of X1 after this kind of a calibrator, right? So in this way, we actually can naturally, generalize this D network based on the training upon of X1 of batch 1 to the inference of batch 2. And that's how the data flow in inference happens. So we have done some simulation study on CyTOF data. So basically, this data is released by Shaham, their paper in Bioinformatics 2017. And basically, we have some peripheral blood mononuclear cells. And these cells, actually, each cell is described by 25 measurements, or we call it 25 features. And for each cell, it has like it can be like in positive class or in negative class, based on whether this kind of a cell has been marked or not. And because this sample is actually processed in 2 different days, so naturally there are 2 batches, right? So if we look at this, just do this kind of visualization to the distribution of all these data points. So each dot here represents a cell, represents a sample. So you can see that these cyan and pink dots actually, they are largely mixed together, but still there's some regions. For example, here you can see this area is dominated by these cyan points, and here is dominated by the pink points. So it means that before this kind of a batch effect removal, in this raw data, these 2 batches are actually they have some very different distributions, right? And after this kind of a batch effect removal, or we call it calibrating, and then we can see that these kind of pink points and these cyan points are indeed mixed together, meaning the distributions of these 2 data sets of these 2 batches are actually coherent and almost the same. And at the same time, if we visualize, just visualize these sample points by using different colors to indicate their class labels, then we can see that before this kind of calibrating, somehow we can roughly separate this orange color, which is for the positive class, and the class 0, which is for negative class, roughly we can do a separation between them, right? But from a machine learning point of view, this kind of a separation is quite nonlinear, because you maybe have to draw a circle or draw some curve to separate them, even in a two-dimensional space, because this is a projected space by I think it's by Disney in the 2D plane. So basically, that is on this 2D plane, you can draw a circle, draw a curve, and to separate these blue points and these orange points. But after calibrating, after batch effect removal, so basically you can draw a straight line, right? Draw a straight line to separate this blue and this orange, meaning that the organization in the space, in the Z space, not in the original X space, in the Z space, actually the organization is improved and has reduced the difficulty to separate the two different classes of the cells. And we have also checked the some quantification here. So before actually we have like 2 patients. So basically for patient 1, we before calibration, it's true that we can do this kind of basically for the first batch, we can do this kind of a cross validation and get a very good accuracy. But if we generalize the model trained on the first batch to the second batch, the accuracy would be dropping. And similarly, we can do this kind of in the batch cross validation, the accuracy is high. But if we apply this model to somehow generalize to a different batch, the accuracy starts to drop. And after calibration, by using the 4 networks combined together by using the system we have proposed, so still for each batch, inside each batch, we can do cross validation, still get very high, similarly high performance here, accuracy here. And the thing that is important is that if we train a model based on batch 1 and we extend it to batch 2, yeah, the performance has been improved if you compare this kind of reading to this reading here. So you might think that this kind of improvement is quite marginal because it is just a simulation study. Let's just see a real example. Try to apply our method to solve this SCLG diagnosis based on mass spectrometry data. So we have recruited with we have collaborated with a local hospital here in Shanghai, a trauma center in Shanghai, and we have recruited nearly 600 subjects, including about 306 SLG patients and nearly 300 health controls. And we have collected all their data by using MADIMOS. So basically, because of the restriction of the scanner, we have to divide all subjects to 3 plates. And naturally, each plate will be a batch. In each batch, we have about 200 people, and the SLE patient almost occupies half of the patient in each batch. So then we can still, try to have a look of this distribution here. And this batch 1 actually points to, we have 3 batches, right? This batch 1 is batch 2. So basically, if we don't do any batch calibration, the 2 batches actually they separate very clearly. And if we train, for example, if we train, and we can see this kind of panel C. Panel C means that in the first batch we have half of the people as SLE patients, and these blue points are for the normal control. And for batch 2, similar story, we have some patients here and some normal control here. So if we train, for example, a classifier based on the data in batch 1, the classifier, a linear classifier, would be very similar to be like this, right? But if we apply this kind of a model directly to, for example, batch 2, it is hard to believe that such a classifier would be, really, generalizable to batch 2, because you have to actually extend this classifier to some unseen domain in this data distribution, right? But with this kind of a data calibration, the first step is that we map from X to Z, from the original space to the latent space, which is coupled between these two different batches. And then you can see that these CM points and these pink points from two different batches, they are mixed together. So originally they are separate, and now they are mixed together, meaning that the calibration has been taken effect, and the batch effect has been suppressed. And then given these pink points here, given these CM points here, for example, batch 1, you can train a classifier. And then if you can apply this kind of a, if we apply this classifier to the second batch, naturally, this kind of a classifier could be generalizable, because now the class 0, the negative class points and the positive class points in the two batches, they are actually distributed very compactly, meaning that we can use a single classifier to solve the problem in two batches. And some quantification here. And similarly, we have like 3 batches, and we can do this kind of inside a batch cross validation. For example, we can do this kind of we can use data from batch 1 to train a model, part of the data in batch 1 to train a model, and to validate on the rest of this batch 1. The performance is quite good, right? For batch 2, and we can do this kind of in-batch cross validation is good. For batch 3, the same story, even before calibration. But the problem is that if we train, for example, a model based on batch 2, and we try to apply it to batch 3, then the performance starts to drop quite a lot, implying that the previously trained classifier cannot work. So, but after calibration, still for each inside batch, each batch, the performance accuracy is good, and the generalization capability has been improved significantly, right? We almost have like a 20% increase for the accuracy here. And the left part is for sample level part, right? But for each patient, we actually can have multiple samples. And here we do this kind of acquisition for, we just repeat 5 sampling for each patient, and we average over these 5 samplings and try to get the subject level diagnosis in the final. And you can see that the kind of performance has been once again stabilized, and which means that the whole method has a good potential to be applied in real scenario. And we have also compared with some of these kind of so-called SOTA methods, state-of-the-art methods. The baseline means that we have no batch effect removal. And combat ratio G, FSVA, they actually commonly use the methods to remove this kind of a batch effect. And ResNet, which was reported in a bioinformatics paper, and basically they're using ResNet to compensate for the batch effect between 2 data sets. The difference is that they have just used this kind of a ResNet to compensate for the distribution. It didn't have classification involved, and it didn't have reconstructor involved. So the performance still doesn't good. Basically, if we train in batch 1 and test over batch 2, the performance is not good. And the last column is for our method. And one item, one module that is very important in our method is about the reconstructor. Reconstructor meaning that we try to recover the original spectrum from the latent code of Z. And this technique is actually widely adopted in medical imaging, especially like in MR image reconstruction, PET image reconstruction. We have all this kind of constraint. And we have also tried to verify whether this module is necessary in our method. So basically you can remove this kind of a reconstructor and try to verify the performance. You can see that, and you can see that the performance has dropped a lot in terms of this kind of accuracy. This is at a sample level accuracy. So once again, it confirms that if we add in this kind of a reconstructor, it's very important to make sure the whole method can work at a more accurate and a more stable status. Okay, so basically, we have been talking about the case for 2 batches, but in fact, or in real setting, usually we have more than 2 batches. We can have 3 batches, as we have seen the previous SLE study, right? And in many more clinical scenarios, I think the batch number can be even can be just more, you know. So in this case, we have investigated the possibility, saying why not just input all these batches in, so you have some source. A source means that in a source collection we have k batches, and in a target collection we have another n minus k batches. So in total we have like up n batches there. The difference is that for source patches, we know the diagnosis labels for each subject sample inside it. So basically, we try to calibrate the batch effect for all source batches and target batches, and then we try to train a diagnosis network by referring to the disease labels in the source batch only, and then we want to make sure this kind of a D network, this diagnosis network, can be generalized to solve the problem in the target batches, right? And we also have done some improvement to the detailed architecture of the network. For example, we have adopted this kind of a REST skip connection, and we have also designed a 2-step alternative optimization skip to attain better numerical stability. And then we can do some validation by using the previously mentioned SLE data set. So still we have 3 batches here. We can train a model by using batch 1 only, and we test over batch 2 and batch 3 simultaneously, right? So if we do this kind of a simultaneously here, if we do this kind of a simultaneous test, we can see the performance actually keeps quite high and quite stable for 2 different target batches. And for the 3, we can swap the order of these 3 batches, for example, by using batch 2 as a source for training, and batch 1 and 3 as a target for inference. Still we can get some very good accuracy here. So as a conclusion, deep learning actually has become a promising way to solve the classification task. It's not just only in genomics or metabolomics. It's actually prevalent in many scenarios. And at the same time, it also provides a means for high-dimensional data mapping. I think at this point actually has been ignored in many studies. So many people just extract the features from some given data, some input data, and eventually by using technique of pooling, etc., and then get a final label. But in fact, in my mind, deep learning actually is a very powerful tool for high-dimensional data mapping. So combine the 2 items above, we have tried some solution to for data mapping and classification simultaneously, and try to apply it to the omics data. And our research is still in the very beginning stage, and still very limited, because we know that there are many clinical applications can be explored. We have some unreported data. I didn't show it in the slides, but we have tried to examining whether the nodule is benign or malign, and we try to figure out whether we have some early sign to screen certain disease, and we are doing this kind of in our lab. And of course, there's some also more recent techniques in the field of artificial intelligence in computer vision, natural language processing, just like the transformer technique or the generative learning technique. And I think this kind of a technique would be very, very useful in the field of metabolomics as well. And finally, thank you very much. And this is Shanghai Tech. I just moved to Shanghai Tech last year. So we have our school is here, and we are building a new hospital here. So we have multiple openings available from faculty to postdoc and to students. And if you're interested, please scan this QR code and you can visit my homepage. And thank you very much.
Thank you very much, Chan. Great talk. And I would like to give the floor to anyone who want to ask question. Biswa, I can see you raise your hand. Go for it.
Yeah, thank you. Great presentation. Couple of questions. One is, did you ever benchmark the method with other existing methods? That's number one. And did you?
Can you repeat it?
Yeah, did you benchmark this method with other existing methods? The benchmarking. Yep.
Yeah, well, the benchmark is that we have, yeah, here, for example, we have some like combat. I think a lot of people are using it. And these are actually a traditional method. And ResNet is a deep learning based method. But in deep learning based method, you have like a different settings. You can do this kind of integration of different modules. And particularly in this method, I think it just use ResNet to try to minimize the distribution discrepancy between two batches. But our experiment shows that it's not enough. We actually can add in more tasks. For example, you can. Yeah, I can just go ahead. For example, we can just solve this classification simultaneously with this calibration, right? Because batch effect removal is not a final target. Our target is actually to deliver a high quality diagnosis or classification.
Yeah, and number two is, did you try with various different analytical platform data sets like LCMS, GCMS, and NMR to be able to test this method?
We have tried, but we didn't try a lot because, as I said, I think I'm a medical imaging person. So for this part of our research, I have done collaboration with my collaborator in another university in Shanghai. And we basically only focus on this kind of muddy mass spectrometric data. And I think it's possible that we can, try more different types of data in the future.
Great, thank you.
Yeah, thank you.
Tian, do you
Hi, Dr. Wang. Can I ask a question?
Yeah, sure.
Have you tried any proteomic data set?
No, we didn't. Yeah, just as in previous question, we haven't yet.
Okay, so I'm asking, have you dealt with any data with missing values? Because one feature of proteomic data is there are quite a lot of missing values. Some of them are biological missing, some are technical missing. And when you deal with a data set with missing value, do you have any advantage?
For this study, we didn't try, but for imaging study, we do have a lot of works. And I think one study that I have done in last 5 years is that we do imaging synthesis. Means that they can generate CT from MR, or it can generate MR from CT. And for that study, we have progressed a lot, and we are trying to for example, like for Alzheimer's disease, there's some PET tracer, which is very specific for A beta and try to diagnose AD as early as possible. but it's just impossible to find this kind of evidence by conventionally in MR. So we have devised a certain way to synthesize PET from MR data, for example, by combining functional MR and by as well as structural MR. And by introducing this kind of additional image synthesis step in the current imaging-based pipeline, we actually can increase the sensitivity and the specificity to for early AD screening. So I think what you mentioned is a very good question, and it is very challenging. We haven't tried this kind of a missing data issue in metabolomics or in this kind of omics data, but it's truly the case that we have tried in medical imaging data, and I think it's a very promising solution, but we still need more efforts on it.
Thanks. I still have many questions, but let's talk later due to a limit of time.
Thank you.
Thank you. Nice to meet you.
Thank you very much.
Thank you so much. Because of the time, we are behind 1 hour. We move on. I'll share my screen for the presentation. Can you see the screen?
Yes, we can.
Yeah. So, Jurek, go ahead and.
Yes, please. Thank you
your
very
turn.
much. So, as you may know, some of the attractions of this meeting, beyond the great science and networking, is as well that we are awarding some presenters with some kind of token. And today, I'm very happy to present that the Journal of Started Biochemistry and Molecular Biology has sponsored as well an award for one speaker. And the wait, the previous slide, please, previous slide. You have spoiled the whole dramatic. So the journal is devoted to the research of signal transduction pathways. It started with steroids, so the name is a little bit misleading, but we are right now publishing as well as a lot of work on vitamin D, lipids, and the metabolomics. The journal is indexed in PubMed and Scopus. It has presently impact factor of 5. There is no publication fee, and open access you can buy with a fee, but this is not required. I am acting as editor-in-chief of this journal. Now, Tusi, please. So I'm very happy to pass this information to Dana, that she is awarded a best presentation award for today. And I am passing my best congratulations to you, Dana. Very well, work very well done. Thank you very much.
Thanks. Thanks, Derek. I'll hand over to Ming to present the best runner-up award. Let me get the slides for you.
Okay, thank you. Susie, can you hear me?
Yeah, we can hear you. And
can you see?
Yeah. Oh, sorry. The slide.
Yeah.
Go for it.
So can you see me on the screen?
Yeah, we can. We can.
Okay. Yeah, good. So yeah, it is my great honor to be presenting this award on behalf of Westlake Omics. We are also one of the sponsors. And yesterday, my colleague Simon said something about a company. Maybe you missed this slide. And the main information here is we are an AI-empowered microbial data company, and we aim at boosting precision medicine and drug research and development by integrating technological innovations in proteomics and multimodal big data. So on the right, there is a vision map. So you can see in the center, there is the brain, and the brain is like our AI. And with this AI as the brain, we can integrate all the players, such as the factory, the data center, the community, into a very smooth running and effective ecosystem. And as one of the sponsors, we are also responsible for disseminating the VPAP in China. So I have a lot of emails and back and forth with Susie, and I have some good news to report to you. Yesterday, we had some live rebroadcast of this VPAP 2022. And in WeChat, we had over 300 online participants, and we had a partner which is called Pushare, and they had two platforms. One recorded 2,300 participants, and the other recorded 1,500 participants. So if we combine the three figures together, we're talking about a viewership of 4,100 viewers from China only. And I think that we can also do something good about publicizing and disseminating those good technology and good science in different parts of the world. So now it is my great honor to present the award. So I'm told that I'd like to present this award to Mr. Amit Yadav from the Translational Health Science and Technology Institute of India. So Amit, are you still online? Yeah, maybe. Yeah, he's online.
Thank you.
Yeah, hello?
Yeah, can you hear me?
Congratulations. Congratulations. And yeah, Westlake Omics is very honored to be presenting this award to you. So thank you.
Thank you. And congrats to Dana as well.
Thank you.
Thank you, Meng. Thank you. Congratulations, Amit and Dana. So moving to our tutorial session. So Biswa, please, my pleasure to introduce Ni for us. You're on.
Yeah, yeah. I think I'm audible.
Yeah.
Yeah. So good morning, good afternoon, and good evening to all. This is my pleasure and honor to introduce our first tutorial speaker for the day, which is Dr. Zhisu Ni. And we had like 20 minutes of talk around how to best pronounce that. So sorry for that. He is one of the upcoming early career researchers doing very well in the field of epilipidomics and lipidomics in the field as such. He is a Carl Gustav Karas of Technical University in Dresden, Germany. Zurek, you would have to excuse me for doing a bad job with the German words. He's been 5 years as a postdoc at Leipzig University as well. He had been an exchange student at University of Vienna. He had a bachelor in pharmacy from China. He had published 18 publications and is a master developer in many lipidomics tools, worked with Maria Fedorova, and has published tools such as Biopan, Lipid Links, LPP Tiger version 1, Lipid Hunter, very active in redox lipidomics and epilipidnet communities. Today he's gonna present on the lipidomics tutorial on LPP Tiger 2, software for prediction and identification of epilipids. Thank you so much, Zhisu, for accepting our invite. The floor is all yours. Thank you so much.
Thank you, Biswa, for the nice introduction. Hello, my name is Zhisu Ni, and Biswa pronounced very correctly. I'm come from TU Dresden medical faculty and the Center of Membrane Biochemistry and Lipid Research from the research group of Maria Fedorova. For over the past many years, we work on the lipidomics field and now in the new term epilipidomics. And we develop so many software and bioinformatics tools. Today, I'm going to present you our tool, LPP Tiger 2, for the prediction and identification of epilipids. So in this 1-hour tutorial, I will give you some basic or crash course about what is the generic workflow of lipidomics or epilipidomics, what is epilipids, and about our new software tools, and give you some hands-on session with step-by-step screenshots to show you how to work with this software. So let's get started. So thanks to the prize award, this talk today about lipids. So we have different lipid classes by this Lipid Maps consortium. We classified them into 8 different classes. there are many different structures within each class. So in the most commonly used database, lipid maps, we have now this number of lipids. Yeah, and they are so diverse in structures, which means they are also having different biological functions in the cell. You have heard this membrane lipids, and then there's also mediator lipids, some like oxalipins, and also other functions as well. You also heard by today's talk about oxidative stress. So in general, in the cell, we have this antioxidant defense, and then this so-called reactive oxygen species, some radicals. And when the cell cannot maintain this balance, or there's too much reactive oxygen species, then they may damage your cell. The molecules within the cell may become oxidized, for example, nucleic acid, lipids, and proteins as well. Over the past decades, there's many studies studying the relation between oxidized lipids and different diseases. The chemistry behind that is in the lipids, we have this unsaturation, and then the reactive oxygen species will attack this allylic position hydrogen next to the double bond and form this lipid radical. And this goes on and on for the chain reaction, and then in the end, it will terminate in different products. For example, peroxide, hydroxide, and also cleavages, and also complex structures like isoprostanes. This can happen to different lipid classes with double bond or fatty acid component, and some of them has been potential biomarkers for some of the diseases. That is quite important, and not only generated by the free radical pathways. Inside the cell, there are also enzymes to catalyze specific reactions to generate site-specific and then stereochemistry-specific structures. And there's also free radical or non-enzymatic reactions for the reactions on different positions as well. For example, the head group. So this is non-enzymatic reactions on the head group of the lipids. In 2019, we come to the idea just to mimic what we have on the DNA. DNA, you have genome, then you have epigenome, you have proteome, you have protein post-translation modifications, like previous talk for today, we have proteoforms, and then we have lipid modifications. So we summarize the enzymatic and non-enzymatic reaction product of lipids into this word called epilipidome. And in the epilipidome, we have lots of different products. I think many of you have taken the medicine ibuprofen, and it will block these pathways and prevent the generation of prostaglandins. And then recently, the oxidized phosphatidylacetamide has been proven its importance in the ferroptotic pathways. And if you're familiar with the cardiovascular disease, you may be familiar with the word LDL, HDL. The LDL can become oxidized. And then you can also find oxidized cholesterol esters. And there are also oxidized triglycerols. So the structures are really diverse. And some of them we know the function, and many of them we don't know the function. In order to know more about the functions of the epilipids, we need to identify them and discover them from the samples. So we need tools and the pipelines to identify them. Here is the generic workflow. So we know today from previous talk, the award winner talks, and then there's lots of things you have to optimize for the sample preparation. And you have to take care of that before you analyze your data, before you obtain your data. But afterwards, there's usually very long time processing of data analysis. And the the solid identification is very important because it will influence all the further analysis and will also correctly represent your sample. So in order to do this, we develop LPP Tiger. Now is the version 2, which can help you to identify oxidized lipids, of course, but also assist you a bit to improve your data acquisition. So as I said, we have so many different structures, and some of them can be isomers. So they are identical in the chemical formula. So they have M or Z, but the real structure is different. You can have hydroxy, epoxy, or a keto group with one less unsaturation. In order to separate them, we need, usually we use LC-MS. So you can separate them in the LC level, but this is not enough. We still need MS spectra to fragment the precursors and really to check the fragments. Then we can see is a hydroxy species or is the epoxy species. So it's very important to use LC-MS for the correct identification of oxidized lipids. And it's also important to obtain more and good quality MS spectra for this low abundance species, because comparing to the unmodified lipids, the epilipids are generally much lower in concentration. We also have the question today about which polarity you have to use. So different lipid classes have their preferences in the ionization. So you can acquire your sample in 2 times, one in negative, one in positive, or you have advanced instruments which can do polar switching. So you have one spectra with both positive and negative information. So this you have to take care before you go to data analysis. This you have to optimize it before you do your analysis. And there are 3 major problems for the identification or general epilipid studies. One is the low concentration. The concentration is like 100 times or even more lower than the unmodified species. There's lots of isomers, and the structure is very diverse. So if you consider the structure diversity plus lots of isomers, so it's really lots of possible structures. So it's really a huge search space, but how big it can be. In one of the reviews, we make some estimation. So if you enumerate all the structures for common lipids, it's around 100,000. But the generic lipidomics paper, if you read some publications, we usually report hundreds or slightly above 1,000 lipids from a real biological sample. If we oxidize them or have some like enumeration over each double bond for all possible combinations of hydroxy, hydroperoxy, keto, epoxy, and so on, and do not limit the number of the modifications, you may end up with 10 to the 10. There's this huge number of enumerated lipids, and all of them located within this smaller range. So around 1 mol/z, you may have 7 million possible structures. This is a crazy number. So, but on the other side, if you use some knowledge-based estimations, say only in the enzymatic or the free radical mechanism generated product, so from this more realistic lipids, which are in some databases, and then you predict the possible structures generated by enzymatic or non-enzymatic reactions, the numbers are much reduced. And then within your real sample, you may identify a subgroup of that in a more reasonable number. So this is our goal for the estimation of the search space. To not search against this number. If you search against this number, you will get really lots of false positives. So you have to reduce your search space to a more or less realistic search space to obtain reliable results. In order to do that, our former student Georgia with us reviewing lots of papers and then construct this polyunsaturated fatty acid oxidation networks. So in the end, we summarize them and classify them into most common modifications from one double bond unit, or some pathway dependent ones, and some also less reported ones. So you can select in our software to which level you oxidize the lipids. And then here comes our algorithm to predict more realistic results. So you take a lipid which you identified, and you take the unsaturated fatty acids, and then you count how many double bond units which you have. You take the first one, you may have oxygen addition, some of the product you selected, or you may have a cleavage here to form a carbonyl terminal or carboxylic terminal. And then if it's not cleavaged, then you go to the next one, and then go to next one until there's no more double bond left. Or there's also another algorithm, which if you have more than 3 double bond units, then you can form this prostand-like ring structures. And then from this one single lipid, you may predict over 100 different structures, but there are only 48 unique MOZ. So there's lots of isomers inside. If you loop this algorithm to all different possible lipids, then you'll get in silico predict epilipidome for your analysis. So with this, we can also enhance or improve our data acquisition. So Palina and Angela propose this new workflow. So we analyze our sample first, just untargeted, simple profiling of unmodified lipids. Then we use our LPP Tiger to take the most abundant lipid species, take the idea that most abundant unmodified lipids with unsaturation are easily or the most substances to be oxidized or modified. And then we take them, and then their product should be more abundant. And then we predict them, and then we perform a semi-targeted LC-MS analysis. And then after that, we can get a better LC-MS datasets with more MS2 of our oxidized lipids, because there's an inclusion list. And then with this better quality, we do identification of oxidized lipids. And then based on the most abundant species, then we can perform quantification analysis in the later round. Okay, so far we partially solved the search space issue by using LPP Tiger. And now comes to the identification phase. In the identification phase, you may think, yeah, we have lots of algorithms for the metabolomics, for the generic unmodified lipids. Yes, there's lots of algorithms already, but the main problems of epilipid study is, first, we don't have a known database for that, and we don't have a big spectral library for that. So many of the algorithms we cannot use for identification of epilipids. Yeah, this is a photo around 10 years ago when I started to do this data analysis of oxidized lipids. You can see here we draw lots of structures by hand or print out some hints. There are really lots of paperwork trying to understand the fragmentation pattern of oxidized lipids. But nowadays, we don't need to do that anymore. So our LPP Tiger software can do it for you, but you still have to understand the basic ideas in order to make correct assignment, your final review of the result and make final decisions. So there are several common algorithms. For example, spectrum matching, when you have a spectral library for that, or you do such a top-down fragmentation of the known structures. And then in this way, you count the M over Z matches. You don't count the intensity of each signal. And the third way is you do some rule-based identification based on fragments or rules. And then you perform this bottom-up identifications. And due to the complexity of oxide lipids, none of them singly solve the issue. So in LPP Tiger, we collect several of these algorithms and add few from ourselves, and then build this 5-score systems. So the generic workflow is like we predict the epilipidome first, and then we load our mass spectra, and then we extract the extracting chromatogram in the software. Then we do a pair of spectra analysis. In the MS1 level, we do a isotope score to check the elemental composition, the isotope distribution. And then based on that, we do our 5-score system to identify our lipids. And then in the end, we will get an interactive clickable report with assigned images. It's like a local web page. You can browse it and then make a final review and then make a final decision. So what is this the isotope score? In the isotope score, you can make this you can check the top 3 isotopes. Yeah. So this is a simple situation. You don't have any co-elution species. You don't have interferences. But in the real world and in the real LCMS, you may not separate your lipids good enough, or there's a common problem that with one saturation you may one more saturation you may introduce 2 more hydrogens, but this will be overlap with the second isotope with 2 carbon-13. So there's overlap of isotope patterns. So you have to clearly assign them and then take the correct part and then to get the real elemental composition. There can also be that some species, for example, phosphatidylcholine and sphingomyelins, they differ by 1. So you may need to solve this one. So LPP Tiger 2 can already solve this by for you, and then you get the correct elemental composition. And then we after you get the elemental composition, there are several ways to go further. Yeah, the first one is the spectrum matching. Yeah? We identify, so we manually assign lots of spectra like this paperwork. So we summarize the fragmentation patterns. So here is one example. So you have the fatty acid loss, fatty acids, yeah, and then there's corresponding spectra. There's also some isomers with different double bond locations. Then you'll see the numbers are different, which is not here. Then if we program the in silico fragmentation with intensities, we get in silico predicted spectra. Then we match it with the acquired spectra. So this is called spectrum matching. So with this, we can say is this structure instead of this, because we don't have this peak, we don't have this peak, we don't have this peak. Additionally, since we have oxidation, so we have a hydroxy group here, so we may have a water loss, and then we can predict all of them. So all combinations which related with this oxidation, so there's more fragmentations, and then we can match them to our spectra. Although some signals are quite low, but this there is support that we have these hydroxy groups. And here is the isomer, so you don't have a hydroxy group, but you have a kettle group with a double bond, with one less double bond. Here in this case, you don't have this signal, you don't have this signal. So if by matching this fragmentation patterns, or we call it a fingerprint match, we do not check the intensities, but we can take the advantage of this low intensity signal region to support the assignment of our structure. Another algorithm is called the rank score, which introduced by Lipid Hunter, or another tool for the identification of unmodified lipids. The basic idea is sometimes some lipid species are very low. When you do lipid profiling, deep profiling to see as many as lipids as possible. Yeah? Some of them may be co-eluted or not separated enough with others, so you have this mixed structure. So you may identify some fatty acids at lower intensities. This also applies for epilipids. And we do not take the absolute intensity, because when you take absolute intensity, it's much lower than others. But we take the rank by the fatty acid signals, because the software is first looking only at the fatty acid and then rebuild the lipid structures. Based on this algorithm, we can rebuild. Yeah, this is the dominant species, because there are two fatty acids and the lysop species. But there's also possibility of the existence of this structure, because we have this signal here, and there's possibility to have this structure. But the dominant species is this one. So we also take this algorithm in LPP Tiger as well. And the last algorithm is we take all assigned signals in the previous scores, and we summarize the intensity. And in the rank score and some scores, we also identified other structure unrelated signals which against the assignment of this structure. Then we perform an algorithm to get the specificity of this structure against this spectra. So this is an example in LPP Tiger 2. So when we take 5 scores altogether, we can get relatively good identifications. So here is the rank score. We labeled all the fragments, major fragments. And then here is the spectra similarity score from the in silico generated spectra. Below is the fingerprint score. These green ones are marked and also are matched ones. There's also specificity score and isotope score. When you summarize them together, you get this quite confident identification to say, yeah, this is a hydroxy group here. So now let's start to have a look of our tool. Yeah, I hope you still follow me without any issues of understanding how this algorithm is going on. If you have questions, you can write me. The software is freely accessible. It's completely free and open source on GitHub. You just go to this link or scan this QR code. And this is our latest release. You just click here, and then you just unpack is a zip file. You just click and run for Windows users. For advanced users, programmers, you can download our source code and do your advanced use for that, and also contribute to our project if you want. And this is the tutorial pack. You can download the tutorial pack. And then here is the later release. You can check from time to time. We may have all current release and previous release document here. So if you download the tutorial pack, we prepare the sample data sets and some sample output for you. And we have this step-by-step user guide. You can open it. It will have lots of screenshots and tell you how to use this software. So if you download our software and unzip it in Windows, and then you'll get a folder. and then you can find this LPP Tiger 2 exe. If you don't find it, you can right click, group by and type, and then you find it easily. So you just double click on it. First, a command window will show up to show where your program is, and then the graphic interface will pop up. So the software is completely clickable, so you don't need to do anything with the programs. So the graphic interface will do it for you. The first thing we want to do is to predict this theoretical epilipidome. There are some files you have to prepare. We already prepared some sample files for you. So the default fatty acid is quite important. These are the common fatty acids with the SMILES representation, basically written where the double bond is in the text form. So we prepared a list of common fatty acids for you with also different double bond locations. And you can also take the other ones. For example, there's a list with 71 different fatty acids with different double bond compositions. Or there's a light list for you to modify by yourself to keep only the major ones from your sample data sets. And you have to prepare using this template a list of files containing your unmodified lipid list. So this will be the precursors here for you to perform an in silico prediction. And you have to make sure all the fatty acid list, all the fatty acids here are in this fatty acid list. So you can perform a check, or you take a broader list, which covers all the fatty acid list here. And then you'll start LPP Tiger. You load this fatty acid list, and use our sample data set. You can load this unmodified. You click here, refine epilipidome by unmodified lipids, and load your file. Here you can select which lipid class you want to oxidize. Here we select PC. You can select multiple ones. Here you can specify the modification site, the maximum number of modification sites. That means here we select 2. That means you may maximum have 2 oxidation on the structure. You can limit the total oxygen. Here we put it to 3, so you can have maximum 3 additional oxygen to the structure. You can have maximum 2 hydroxy groups and maximum 1 keto, or maximum 1 hydroperoxy. With this configuration, you may have by the maximum 1 hydroxy and then 1 hydroperoxy. So you will reach 3 additional oxygens to the structure. And then you select a place to put this output file. It's a JSON format, which can be opened by bioinformaticians, but it will be also needed for the later use in the LPP Tiger. And then that's easy. Then you run this prediction. Just click that. You can monitor what is going on in the terminal, and then you'll see the task is finished. Yes, task is finished in the graphic interface as well. You can see if you do not limit the fatty acid list, the number is above 10,000. If you limit the fatty acid, the lipid precursors to the ones defined in the unmodified lipids, the result will reduce to 500 something. Yeah? So it's really limited, and then sample specific epilipidome, which is more reflecting the possible epilipidome space for your sample. Yeah, this is the result JSON file. You can open that with some editors or you ask your bioinformaticians to take more look at it. It has structures and more that, many information inside. and you can freely reuse it in other bioinformatic pipelines. And after you generate the prediction, and then you can come to here. So as we said, you can optimize the data analysis, data acquisition by using inclusion list. So LPP Tiger can help you to prepare this list. So you take again this unmodified lipids list, and then you load this predicted epilipid space, the JSON file, and then you choose where to save it, and then you simply press this generate inclusion list button, and then very fast it will generate, because we have here only PCs. So in this list, the cholesterol esters and TGs are not generated. It's fine like that. And then this is the output file. You open it. You will see a list of big table. You have lipid names. You have the neutral formula, the neutral mass, the charge, and then the charge formula, and so on. You can load it to different instruments to generate this inclusion list. And we also prepare for you the oxidized fatty acid with the name, formula, and neutral mass. With this, you can generate the MRM transitions or PRM list for the LCMS acquisition. So you can freely reuse it for your data acquisition as well. And then after the data acquisition, we can make the data analysis. So you download your file. Here you download the Proteo Wizard, which is a common tool to convert commercial software, commercial LCMS datasets to the common free format called MZML. You download the Proteo Wizard. There's a tool called MS Convert. You start that, you load your files, and then you select output format to be MZML. And then you select here the 32-bit. This is just to shrink down the file size. If you select 64, it's also fine, just the file size will be bigger. And then you click start. It will convert your raw files into MZML files. We prepare some MZML files for you in this tutorial as well. And it's very important that you keep using the same version for your one project, because the Proteo Wizard team is constantly releasing new versions, but it's better to keep the same version for the same data set. So you convert always using the same software without having issues during the conversion. So now we have the epilipidome predicted. We also have the spectra converted. Now we can load into our software. So you select here the lipid identification tab. You load the MZML files here. And there are many informations you can select. These are technical informations you have to know from the data acquisition side. Yeah? So you have to set the tolerance in the MS1 and the MS2 level. And then you have to define the retain time range of your targeted or the lipid you want to look for. For this file, we have 20 minutes negative run for phospholipids. So we set here maximum to 20 minutes. This precursor intensity means the precursor should be at least this intensity to consider as a real meaningful signal. You can also search a broader range or narrower range of precursors. The selection window, you have to ask the people who acquire the data, or you know it by yourself. It's the LCMS method dependent. So the instrument will select a range collecting ions for fragmentation. And this is, you have to consider it. And then you have to define the score threshold. You have the total score, and you have the isotope pattern scores, the rank scores, and then you can set here. So consider peak only at least 1% relative intensity. You can decrease it or increase it based on how the noise level of your spectrum. Then you load this predicted epilipidome here. And then you have to set two output. One is the report folder. In this folder, we'll store this interactive output with images. And then the other one is an Excel output table, which you can have an overview of all the output in Excel table format for later processing. You can also select here if you want to see what is going on within the program, or you want to reuse some data, or your bioinformatician is just curious. You can dump all the raw data as a JSON file, so you can load it in the program to see what is going on. All the assignment scoring details are saved here. You can also adjust the figure quality. You can select 300 dpi for higher quality, or even submit as supplements for journals. Then you just click here, hunt lipids, and then it will become gray here, and then says it's hunting lipids now. And then the command line window will loop some information. So you can say some image is saved, some precursors are identified, some isotope check is passed, so you can monitor how the program is running. Yeah, this window is black, but our tool is not a black box, so you can see fully transparent what is going on behind that. And then when the run is finished, then the interface you can see, it's finished. And then we can check our results. So here is the results folder. You can see the Excel table, and then here is the local website you can open by different web browsers. And then you'll see this local website. Here are the major parameters. Here is the results table. You can click on each of that. It will show you the corresponding assignment images. Let's have a more detailed look of this so-called 6-panel figure. So here you can see the extracting ion chromatogram, where the MS and corresponding MS is taken. And then you can see here the full MS spectrum on the full MS level. And then you can see here the isotopes. So you can see the isotope pattern, and then corresponding isotope score. And now here on the right side are all MS2. Here is the full MS2 with all 5 scores, and then the assignment with the total score here. And then here are the zoom for below 400 or above 400 for you to look more closely the assignment. Sometimes you'll get different isomers, both identified from the same scan. So you see here the scan ID is the identical, so it's the same spectrum. We identify both. There are 2 candidates with different scores. Then you can review it manually. So here on the left side is a hydroxy assignment. You can see here is already assigned. So the fatty acid 22,4 with 1 hydroxy with the loss of 1 water. It's also here, you see it more clearly with the MOZ written here. On the right side, here is marked as red, and it says here is identified as the fatty acid 22,4 with hydroxy as with the water loss. So it's marked here. That's why it has a lower score. Then you, after manually review it, just quick look for a few seconds, you can say, yeah, this is the correct identification. Yeah? This one you will remove from the result table. Yeah? So you can easily review it and then select the best output. So the previous version of LPP Tiger, we take really lots of time to run it, but now we improve our speed a little bit. But in 2019, we have this review together with Professor Laura Gracci from University of Perugia. We have this idea that we use the high throughput with low throughput methods. So in the high throughput, we have the lipidomics data analysis very fast, and then we can pre-filter some potential oxidized lipids. And then we select only these oxidized lipids to LPP Tiger for more deep analysis. So now in LPP Tiger 2 recent release, we finally make it possible. So with LipoStar 2, you can do lots of analysis, and then you can process hundreds of raw files, and then perform lipid quantification and normalization. And then you can export the output to LPP Tiger 2, which normally run single file each time. But we do it more deeply with our 5-score system. But after identification from LipoStar data, we can export again, and LipoStar retake the identification from LPP Tiger 2 inside LipoStar 2. So you can continue your further analysis for quantification inside LipoStar 2. You can feel free to test this. We are still working on a tutorial for that. So now let's see the complete workflow again. So we have LPP Tiger 2 covering help you to do the LCMS analysis and the data analysis. We also have other tools over the past years developed in our lab for the identification of unmodified lipids. That's LPP Tiger Lipid Hunter 2. And we have these lipid circles, which we can visualize lipidomics data in the circles plot. We also invest time to develop our lipid links tool, which is now hosted by Lipid Maps. So in this way, we help to solve the general issue that the lipid had different names, and in different databases, we have to use different names. And then it's also really nice opportunity for this nice event to learn different omics data technologies. And then we all talk about multi-omics data integration. In the end, we also want to merge our lipidomics data with other omics data and perform some advanced network analysis. This is the overall ideal workflow. Yeah, as we said, there's already lots of tools, not only from our lab, but also from different labs. And it may be complex for researchers in the lipidomics field. So together with Lipid Maps, we developed this lipidomics tool guide. Now it's really accessible on the Lipid Maps website. I'll give you a quick introduction of that. So this is the Lipid Maps website. You click here, mass spectrometry tools, and then you find lipidomics tool guide here. When you click that, you come to this workflow page with graphic workflow of each steps, or you can click view all tools. You can see the list or browse different tools. Here we collect all the free available tools for academic people with interface. So you don't need any programming skills to use any of these tools, and you can build your own pipelines. This is a huge collaborative work initiated by Lipid Maps. We ask all different software developers of all these tools to contribute, and then we build up this nice interactive website for you to build your workflow. Here is an example workflow. You can take Lipid Maps structural database, the most commonly used ones, use LipoStar for identification and quantification, and perform some data analysis. You can take the metabolomics workbench to store your data, and then you can use other tools like LionWeb or Pathways for advanced analysis. You can use lipid links to convert your IDs in the standard forms or exchange between different tools as well. If you click one of the tools, for example, lipid links here, you'll come to a description web page. It will show you what the tool can do, some basic information, how to use it, where to get it, what it requires you to prepare, and so on. Here, you can have the task specific information, more in details, what this tool exactly can do. You can also go to LPP Tiger. You can see LPP Tiger, it can do spectral matching, and it has decision rules and so on. You can browse this website on Lipid Maps and then build your own workflow. And in the end, I would also like to introduce our nice epilipid net. It's based on the idea of epilipidome. We have this joint network founded by the course actions from European Union. It's a collaborative network connecting people working on epilipidome. This is basically mainly EU central base for close interaction and some fundings, but we also accept other people from Asia, US for joining our network. We have collaboration opportunities. We also have from time to time Zoom meetings to show our work and discuss some further plans for the field. There are several work groups. There are also meetings for each group from time to time. And in the work group too, we spend our time to collect our work together and then review the latest epilipidomics software. This is already submitted, and then hopefully it will be online in January 2023, so January next year. And you can follow us on Twitter to get more information about our coming events, and also you'll get notified when this review paper is online. So that's the end of my talk. I would like to thank my supervisor, Maria Fedorova, supervised me from master time doing oxidized lipids. Until now, we are so far many years in this field. And we also have other great team here to work all together on the lipids. And I would like to thank EpilipidNet as well. Also, special thanks goes for Lipid Maps to organize this nice Lipid Maps tool guide and host our tools for lipid links. You can feel free to make screenshot or scan this QR code to know more about our tools. And also we have some other tools also free available on GitHub. That's the end of my talk. Thank you for your attention. I hope you will like our tools. And then if you have any questions, please ask me. Thanks.
Thank you so much, Neem. Such a great presentation and great tutorial where the bottleneck for the lipidomics. Bisa, do you have any question? Or can I jump with one question first?
Yeah, go ahead and take the question in the chat.
Thank you. You show that most of the lipid species based on a mammalian. Do you have any interest to expand that for the plant lipids like the MGDG or galactolipids, those type of?
Yes, actually in this epilipid net, we have new some subgroups. Some professors in Europe, they have plant lipids and also fungi lipids from fungi. Yeah. They are also very different from the mammalian lipids. There are some subgroups. Just feel free to join us or get contacts. We have, once you join, you will have this membership area. You can see the expertise of each member. You can also fill yours to search for collaborations and discussions. We also, it's very important also to expand our knowledge. And you can also feel free to contact lipid maps if you want to add more lipids. Now in the lipid maps, I don't have the latest screenshots. Here you go to lipid maps, then there's a new lipidomics forum, and then you can suggest to add more lipids. If you identify some new lipids, you supply the paper references, and then they will try to add in as soon as possible.
It's great. No, I'll contact you with the more
Yeah, thanks.
information.
Yeah, the EpiLipidNet is completely free. You can join, and no matter where you are in the world, you can join. And then we have these online Zoom events from time to time.
Yeah. There are more ideas in my head, so I'll come
Yeah,
back.
yeah,
Thanks.
that's welcome.
Quick question. Great set of tools. Looking forward to use many of those I have used all of them in the past. Question is, do you have recommendations of controlling for false discovery rates? FDA as per those predictions. If anything
The false discovery rate in the epilipid domics is always questionable. It's hard to use decoy data set or something to really to measure the false discovery rate, because there are so many isomers. Even if we use 5 different scores, maximum we can get is like 3 or 6 differences, sometimes 1 differences in the score. But as a human, if you read this 6 panel images, you just need 5 seconds just to say this is the correct one. Yeah? That's why we use we spend lots of effort to build these 6 panel images, and with this local website, you just click, and then you just keep or delete the corresponding ones in the Excel template. You can get the correct ones. Because really, with all these 5 scores, yeah, if you look only the scores, it's nice, but if you consider isomers, then it's difficult. Maybe later with machine learning tools or retained time mapping and Kendrick mass plot, if we summarize all together, we can improve this and increase the difference between these two. And then maybe one day it will be only identified as this one, but it's still a developing field. We will try our best effort to improve the results in the coming years. But it's still our goal, let's say.
Got it. Yeah. Thank you. Yep.
Any more questions for me?
I see from the chat, let's say, why do we need in silico prediction of top N lipids? Couldn't we do that in LC? This is maybe I'm explaining a bit too fast. Here it's not we select top N, yeah? It's not the targeting methods, but it's like you select the top N most dominant unoxidized lipids, and then you predict the oxidized lipids. So let's say if you have, let's say I have these slides here. Let's say in this list, the PC we submit is like less than how many of that? In this PC, we have only this, let's say 13, 15 PC, yeah, phosphatidylcholanes, unoxidized one. We predicted it will be like 200, I think, or more oxidized product, depending on your settings. Then with this, then you can upload to your instrument to set up the method. But if you have not only PC but more, you cannot do that just by manual calculation or just directly submit to the instrument, because there are also additional issues. For example, some PCs, they are most dominant in the formate adduct or acetate adduct. Some of them, if you have carboxylic terminals, carboxylic acid terminals, then they become deprotonated. So the software also do it automatically for you. Don't need to just take it. And if you do manually, then you have to think and calculate differently. It's very important. It's also for This is a special issue for the oxidized lipids that the adduct, the most dominant adduct type may change. And this you have to consider. Yeah. Another is what are the advantages using LCMS to detect epilipids compared to GCMS? The GCMS is very popular for identification of fatty acids and oxylipins, as to my knowledge. Yeah, if you identify small molecules, it's usually no problem. There are established database and methods. But when we consider diverse or complex epilipids, as we say here, its function with connection to the full phospholipids, or they have these complex structures like here and here. Is you either need derivatization or to measure on GCMS, since many of them are not volatile. There's some smaller molecules like HNE, they are volatile, or they can be easily derivatized, then you can use GCMS. But I think the most, according to our epilipid experience, the most common one is to use LCMS, because you can basically do label-free. And each step, when you do derivatization or sample pre-treatment, you may add oxidation. So this previous talk about the lipid extraction, it's very important to, if you want to do epilipid domics, and then you have to add antioxidant in the very early stage of your sample preparation, for example, BHT, to prevent any artificial or in vitro oxidation from your sample. Yeah? We have one publication, you just put your PAPC, some polyunsaturated fatty acids, some unoxidized phosphatidylcholine in the room temperature, open the valves, and then stay for a while, then you come automatic oxidation. So you have to really add in the antioxidant in the sample preparation to prevent any like artificial oxidation during your sample preparation. Yeah. Any more questions?
See any in the chat? Because of the time, yeah. Unfortunately, yeah. So we better keep moving, but unfortunately, Associate Professor Wilson Ko won't be able to present because of the timing. He had the urgent meeting to attend, so we have to finish the virtual podium for today.
Yeah, thanks for inviting us, and
yeah,
thanks.
Thanks, May. We really appreciate. So I would like to thank all the speakers today and the chairing, helping, and especially lightning talks. Really, really interesting talks. Cannot ask anymore, and we are running out the time again. But Professor Wilson's tutorial will be coming up later. Not with the virtual podium, but we will organize something separately, and we'll let you all know when it's going to happen. So please join, because really sad to miss that tutorial today. My apologies. Anything to add, Jurek or Biswa?
Oh, I would like to add my opinion as a human-oriented metabolomic guy. I am learning so much during this all presentations. I would never go to the dedicated software-oriented symposium, but we have specialists talking about the topics which are of interest to me. So this is how this online meeting is supporting the networking and the knowledge. So I'm looking forward to tomorrow. And I would like to thank all audience and speakers for great contributions.
Thank you, Jared. Biswa, you want to say anything?
No, all good, and great job by the lightning talks and the tutorials. I'm sorry to miss out on Professor. Hopefully we can at some other point then. But yeah, a very learning experience. Looking forward to exciting day tomorrow too.
Yeah.
Thank you all.
Okay. Thank you very much, and see you tomorrow. And if you miss, you can join the stream tonight. Yeah, in a few hours time. Tianan, you want to add something? Sorry, I haven't seen.
That's fine. It's a great meeting. It's a bit pity that Wilson cannot talk today, but let's wait for another tutorial for the multiplex analysis.
Yeah.
Thanks a lot.
Bye-bye.
Bye.
Bye-bye.
So you stop the recording?
About